In 2017, researchers analyzed crustaceans from the floor of the Mariana Trench and found industrial chemicals in their bodies at concentrations higher than animals from polluted coastal harbors. These creatures had never been near a coastline. They lived 11 kilometers below the surface. Today, we uncover the most terrifying creatures hiding in every ocean zone. Section 1. The Intertidal Zone. Box jellyfish. The intertidal zone is the strip of coast between the high tide mark and the low tide mark. It is ocean half the time and open air the other half. Rock pools, sandy flats, mangrove edges, shallow reef shelves all fall within it. It is the most visited part of the ocean by any measure, the part people wade through barefoot on warm afternoons. It is also the primary hunting ground of Chironex fleckeri. Chironex fleckeri is a box jellyfish, named for the roughly cubic shape of its bell. The bell reaches up to 30 centimeters across. From each of its 4 lower corners hang up to 15 tentacles, giving the animal up to 60 in total, each capable of extending to 3 meters in the water. The tentacles are almost transparent, close to invisible against moving water, and they are covered end to end in structures called nematocysts. Each nematocyst is a pressurized capsule containing a coiled hollow thread tipped with venom. When a tentacle contacts skin, chemical compounds on the surface of that skin trigger the nematocysts to fire. The thread uncoils and punctures the outer dermal layer in a fraction of a second, injecting venom directly into the tissue below. A single significant contact with one tentacle can involve hundreds of thousands of nematocyst discharges at once. The venom is a complex mixture targeting multiple biological systems simultaneously. One of its primary mechanisms involves disrupting the membranes of red blood cells, causing them to release potassium into the surrounding blood. Potassium is essential to the electrical signaling that keeps the heart in rhythm. A rapid spike in blood potassium levels interferes with that signaling and can push the heart into dysrhythmia. In a severe sting involving significant body surface area, cardiovascular collapse follows. The documented timeline from initial sting contact to cardiac arrest, in some cases, is under 5 minutes. Pain at the contact site is immediate and severe, described in clinical literature as among the most intense pain associated with any marine injury. But in a serious envenomation, the systemic collapse overtakes the local pain quickly. A distinctive frosted or ladder-patterned welt marks the skin where the tentacle dragged, and it persists long after the sting itself. In fatalities, these marks have served as the primary diagnostic indicator of the cause. No other marine animal is formally classified as more venomous by measured toxicity per volume of venom delivered. That classification requires context. The actual outcome of any given contact depends on how much of the tentacle surface touched the skin and for how long. A glancing brush with the tip of a tentacle delivers far less venom than full entanglement across a large area of the body. Most encounters between humans and Chironex fleckeri do not end in death. But the encounters that do end in death do so with a speed that is documented and not disputed in the medical literature. There is no extended symptomatic window of the kind seen with many other venomous animals. In a worst-case contact, the interval between the sting and the loss of cardiac function can be shorter than the time it takes to reach shore from open water. The species was not formally entered into the scientific record until 1956. A physician named Hugo Flecker practiced in Cairns, in far north Queensland, and spent years collecting specimens from the region's waters and documenting deaths along the coast that he believed were attributable to box jellyfish stings. His published notes from the 1940s described the pattern of injuries and the speed of death in cases he had attended or investigated. The creature killing people had no scientific name during those years. Ronald Southcott, a South Australian naturalist and physician, formally described the species in 1956 and named it fleckeri in Flecker's honor. The naming came years after the deaths Flecker had been documenting, meaning a medically significant and lethal species was operating in populated coastal waters without a formal classification for more than a decade of recorded fatalities. The primary geographic range runs through the Indo-Pacific coastal zone, northern Australia, particularly Queensland and the Northern Territory, Papua New Guinea, the Philippines, Vietnam, and surrounding coastal waters. Within that range, the animal's presence follows water temperature. In northern Queensland, the stinger season runs roughly from October through May, aligned with the warm, wet season. During those months, Some beaches post closure signs and others install stinger nets, which are large mesh enclosures anchored offshore to create a partial barrier between swimmers and the open water. The nets do not span the full water column and cannot exclude every animal that moves through, but they significantly reduce exposure inside the enclosure. Outside the nets, full-body Lycra stinger suits are the recommended protection for anyone entering the water during season. The visual system of Chironex fleckeri has drawn sustained research interest because it is genuinely anomalous. The animal has 24 eyes, arranged in 4 clusters, called rhopalia, positioned around the bell. Each cluster contains 6 eyes. 2 of the 6 in each cluster are structurally complex, with a cornea, a lens, an iris, and a retina, the same fundamental components as a vertebrate eye. The other 4 in each cluster are simpler pit-type photoreceptors that detect light intensity and direction without forming a focused image. The full arrangement gives the animal visual coverage across nearly the entire sphere around its body. In controlled laboratory experiments, box jellyfish have been shown to navigate around obstacles and to orient themselves toward the shaded water beneath overhanging vegetation. The leading interpretation is that the image-forming eyes detect the canopy of mangrove trees above the waterline and steer the animal toward the roots below, where small fish and shrimp concentrate in sheltered conditions. The behavior is repeatable and documented. What remains unresolved is the mechanism behind it. The animal has no brain. There is no centralized neural structure receiving and integrating the visual signals from 24 eyes and generating coordinated motor output. Instead, Chironex fleckeri has a diffuse nerve net distributed through the tissue of the bell. How that net produces consistent, directed behavior from distributed sensory input without a processing center is a question that has been studied across multiple research groups and not yet fully answered. The behavior exists. The substrate producing it does not resemble any system most people would associate with navigation. Unlike most jellyfish, which move where currents carry them, box jellyfish are active swimmers. Chironex fleckeri contracts its bell in rhythmic pulses to propel itself and can reach speeds that researchers have measured at around 2 meters per second in short bursts. It moves toward prey rather than drifting into it. Hunting involves swimming through shallow water with tentacles trailing, making contact with fish or shrimp, immobilizing them rapidly with venom, and then retracting the tentacles to bring the prey toward the mouth on the underside of the bell. The whole sequence, from contact to digestion beginning, is efficient enough that the animal can capture and process multiple prey items in a short period when food density is high in the water. Documentation of deaths in Australia is among the most thorough in the region, partly because the medical and emergency infrastructure there has been in place long enough to generate reliable historical records. Estimates of confirmed deaths attributed to box jellyfish in Australian waters since systematic recording began run into the dozens, with some researchers placing the total over the 20th century higher. Researchers have also noted that the documented toll in the Philippines and parts of Southeast Asia is likely greater in absolute numbers, with some estimates placing annual fatalities in the Philippines alone at between 20 and 40 deaths. Reporting systems vary significantly across the region, and deaths from marine envenomation in rural coastal communities are not always formally categorized in ways that enter national health records. The result is that the broader human cost of Chironex fleckeri across its full range is probably underrepresented in the published literature. First aid protocols for box jellyfish stings have been revised and standardized through research over the past several decades. The current guidance from marine envenomation specialists calls for dousing the contact area with vinegar as quickly as possible after the sting. Vinegar is acetic acid, and its role is specific. It does not neutralize venom already injected into the tissue, but it deactivates unfired nematocysts still adhering to the skin. A person removed from the water with tentacle fragments still on their skin continues to receive venom injections from those nematocysts until they are either deactivated or removed. Rubbing the tentacles off, which is the instinctive response to the pain, triggers mass discharge from the remaining nematocysts and substantially increases the total venom dose. After vinegar application, fragments can be removed without direct skin contact. Cardiopulmonary resuscitation should begin immediately if the victim has lost cardiac function and the documented survival cases from severe stings consistently involved CPR being started quickly and maintained through transport to hospital care. An antivenom has been available in Australia since the 1970s. Its role in severe envenomations has been the subject of ongoing debate in the medical literature. Some research has suggested that by the time cardiovascular collapse is underway, administering antivenom may not reverse the damage already done to the heart's electrical system. Other clinical reports have supported its use in reducing severity in moderate stings and in cases where it is given rapidly after contact. The debate is not resolved, and treatment protocols differ between institutions. What is not debated is the priority of resuscitation over any other intervention in a victim who has lost cardiac rhythm. Research into the venom mechanism has continued to advance. Teams in Australia and elsewhere have identified specific proteins in the venom responsible for forming pores in cell membranes, the pathway by which potassium is released from red blood cells and enters the bloodstream. Characterizing that mechanism at a molecular level has clarified the biological chain of events leading from tentacle contact to cardiac failure, and it provides a theoretical target for drugs that could interrupt the process before collapse occurs. No such treatment has entered clinical use as of the most recent published literature, but the pathway is understood with more precision than it was even a decade ago. The intertidal and nearshore waters of northern Australia also host a related cubozoan that operates in a very different way. The Irukandji jellyfish, most commonly the species Carukia barnesi, is small enough to pass through stinger nets. Its bell is roughly the size of a thumbnail. The initial sting is often barely noticed. What follows, between 5 and 40 minutes later, is a syndrome named after the Irukandji people of coastal Queensland. Severe pain radiating through the back, chest, and limbs, nausea, sweating, a dangerous rise in blood pressure, and a documented psychological symptom described in medical case reports as an overwhelming conviction that death is imminent. The syndrome is caused by a massive release of catecholamines, stress hormones, triggered by the venom. Deaths have been attributed to Irukandji syndrome, primarily through hypertensive crisis or cardiac complications arising from the hormonal surge. The species, Carukia barnesi, was named for Jack Barnes, a physician who in 1964 set out to prove that a tiny jellyfish was responsible for Irukandji syndrome. Barnes captured a suspect specimen and, in a documented episode that would not pass any modern ethics review, stung himself, his 9-year-old son, and a local lifeguard with it. All three developed Irukandji syndrome and were hospitalized. The experiment confirmed the cause. Barnes wrote it up and submitted his findings. The species now carries his name. The intertidal zone is not a place most people associate with serious danger. It is the accessible margin of the ocean, the part with gentle surf and shallow water, and rock pools full of small things. That familiarity is part of why Chironex fleckeri causes the harm it does. The animal is not aggressive. It does not pursue humans. Contact happens because a person moves through water where tentacles are already trailing, invisible in motion. The encounter is accidental from both sides. What follows depends on how much surface area was involved, how quickly the person reaches shore, and how quickly the right response begins. In the documented survivals from severe stings, those factors aligned. In the fatalities, at least one of them did not. Before we move on, if you're enjoying this analysis, please consider subscribing. It helps the channel tremendously. The next section moves out past the shore and into the open sunlit water, where a predator tracked by satellite across entire ocean basins has a sensory system far stranger than the popular version of it suggests. Section 2. The Sunlit Zone. Great White Shark. In September 2012, a research team working with the ocean tracking organization Ocearch tagged a large female great white shark off the coast of Cape Cod, Massachusetts, and named her Mary Lee. She weighed just over 1,600 kilograms and measured nearly 5 meters from nose to tail. A satellite transmitter was attached to her dorsal fin. Over the following years, every time her fin broke the surface long enough to ping a satellite, her position was logged and made publicly available. She traveled up and down the eastern seaboard of the United States, moved out into the open Atlantic, turned south toward Florida, and then disappeared from the record entirely when her transmitter stopped pinging. In the years she was active, her movements drew more public attention to white shark migration than decades of prior research had managed, not because the science was new, but because a named, trackable individual made the scale of it legible. A single shark, moving across thousands of kilometers of open ocean, surfacing and diving and hunting, across a range that dwarfed any reasonable intuition about where a fish might go. Carcharodon carcharias is the white shark's full scientific name. The common name, great white, comes from the coloration of the underside of the animal, which is a pale off-white against a gray or gray-brown dorsal surface. That two-tone pattern is called countershading, and it serves a functional purpose. From below, the pale belly blends with the lit surface water above. From above, the dark back blends with the deeper water below. The coloration makes the animal harder to detect from both directions simultaneously, which is relevant because the white shark approaches prey predominantly from below and behind. Adult females are larger than adult males. Confirmed female lengths above 6 meters have been recorded, though the extreme upper end of size claims requires caution because measuring a live or recently dead large shark accurately is difficult, and historical records were not always methodologically rigorous. A large female filmed near Hawaii, given the name Deep Blue by researchers who documented her, was estimated by experienced observers at around 6 meters and was visibly pregnant at the time of the encounter. Males typically reach 4 to 4.5 meters. The largest reliably measured specimens in the scientific literature fall in the 5 to 6 meter range. Body weight at those sizes exceeds 2 tons. The teeth are serrated and triangular, designed for cutting through flesh and bone rather than holding prey. The white shark does not chew. It bites, shakes its head laterally to sever tissue, and swallows. Multiple rows of teeth sit behind the front row in a conveyor-like arrangement. When a front tooth is lost or worn, the tooth behind it rotates forward to replace it. A white shark cycles through thousands of teeth over the course of its life. Each tooth is individually shed and replaced, and shed teeth are the most commonly found white shark remains in the fossil record, because enamel preserves where soft tissue does not. The sensory system of Carcharodon carcharias has been studied extensively and turns out to be considerably more layered than most public descriptions of it convey. Vision is one component. White sharks have large eyes with a tapetum lucidum, a reflective layer behind the retina that amplifies available light in low visibility conditions, similar in function to the same structure in cats. When attacking prey from below in bright surface light, the shark rolls its eyes back into the skull to protect them during impact. The eyes are not fixed forward in the way a human's are. The field of view is wide and positioned to give awareness of what is happening around and behind the animal, not only in front of it. Smell is another component, and the olfactory capability of sharks has been widely overstated in popular accounts and equally widely understated in skeptical corrections of those accounts. The real picture sits between the two. White sharks can detect certain organic compounds, including components of blood, at very low concentrations in water. The detection threshold for some compounds is in the range of 1 part per million or lower. The shark does not smell blood from miles away in any simple sense, because scent in water does not travel in a uniform cone but disperses along current pathways and breaks up over distance. What the shark is actually doing is following a chemical gradient, moving in the direction of increasing concentration. That is a fundamentally different process from smelling something from a fixed point, and it is more accurate to describe it as gradient navigation than distance detection. The ampullae of Lorenzini are perhaps the least intuitive part of the system. These are small gel-filled pores distributed across the snout and lower jaw. They detect weak electrical fields in the surrounding water. All living animals generate electrical fields through the electrical activity of their muscles and nervous systems, including the heart. A beating heart produces a detectable electrical signal. The ampullae of Lorenzini allow a white shark to detect that signal at close range, meaning an animal that is motionless, visually concealed, producing no chemical signal, and making no sound, can still be detectable to a nearby white shark through the electrical field generated by its own cardiovascular function. This capability is used in the final approach to prey, when other sensory systems may have already guided the shark close. It is also likely involved in the shark's ability to navigate using the Earth's magnetic field, which generates weak electrical currents as the shark moves through it, though the specifics of how white sharks navigate across ocean basins remain an active area of research. Regional endothermy, sometimes called mesothermy, is a physiological trait that separates the white shark from most other fish. The animal is not cold-blooded in the way a cod or a herring is. Through a system of countercurrent heat exchange in the blood vessels, the white shark maintains the temperature of its core muscles, brain, and eyes several degrees above the surrounding water temperature. In cold water, this gives the animal faster muscle response and sharper neural processing than a truly cold-blooded animal of the same size would have. It also allows white sharks to operate across a wide range of water temperatures and to make deep dives into colder water without the performance penalties that would affect an ectothermic animal. Tracking data has shown white sharks descending to depths exceeding 1,000 meters on some occasions, well below the sunlit zone, before returning to shallower water. The hunting behavior documented most extensively involves the approach from below toward prey at the surface. Off the coast of South Africa, particularly in an area of False Bay near a seal colony, white sharks were documented and filmed attacking Cape fur seals from directly below at high speed, breaching clear of the water in the process. The attacks are fast enough that the initial strike is often decisive. The shark accelerates from depth, hits the seal at or near the surface, and the impact alone is frequently sufficient to kill or incapacitate the prey before the shark circles back to feed. Not every attack follows this pattern. White sharks also scavenge whale carcasses, feed on fish, and take other sharks. The dramatic breach attack on seals represents one context-specific hunting strategy, not the only one. For much of the 20th century, the lifespan of Carcharodon carcharias was estimated at around 30 years. That estimate was based on growth ring counts in the vertebrae, similar to how tree rings are counted, but the methodology was uncertain because it was not clear whether each ring represented 1 year of growth. A study published in 2014 applied bomb radiocarbon dating to white shark vertebrae, using the atmospheric pulse of radiocarbon from nuclear weapons testing in the mid-20th century as a fixed chronological marker. The results revised the lifespan estimate substantially upward. One male in the sample was determined to be 73 years old. Female lifespans were estimated to potentially exceed that. The revision had significant implications beyond simple natural history interest, because lifespan is directly connected to reproductive rate and the speed at which a depleted population can recover. Females do not reach sexual maturity until around age 33. Based on the same radiocarbon research, gestation has not been directly observed in the wild and is estimated to last approximately 12 months, possibly longer. Litter sizes are small, typically between 2 and 10 pups, and the interval between pregnancies is thought to be several years. The combination of late maturity, small litter size, and long interbirth intervals means the white shark reproduces slowly even by the standards of large predatory fish. A population reduced by fishing pressure, bycatch, or other mortality cannot rebuild itself quickly. The IUCN currently lists Carcharodon carcharias as vulnerable on its red list of threatened species, and population estimates, which are difficult to produce reliably for a wide-ranging oceanic predator, have suggested that total global numbers may be in the low thousands. The relationship between white sharks and humans has been extensively documented through the International Shark Attack File, maintained at the University of Florida, which has compiled records of shark encounters globally for decades. White sharks are responsible for the highest number of unprovoked bites on humans of any species. That statistic is frequently misread. The total number of fatalities from all shark species combined in a typical year globally is around 5 to 10. White sharks account for a disproportionate share of those, but the absolute numbers are small relative to the frequency of human presence in the water. The shark's involvement in bite incidents appears, based on behavioral research and the nature of most encounters, to often be investigatory rather than predatory in the hunting sense. White sharks bite novel objects in their environment. A surfboard from below, in low-visibility water, has a silhouette with some similarity to a seal. Most survivors of white shark bites report a single contact, after which the shark did not return to feed, which is not consistent with the behavior of an animal treating a human as preferred prey. Aggregation sites where white sharks gather in reliable numbers have been studied intensively. Guadalupe Island, off the Pacific coast of Baja California in Mexico, hosts a population of white sharks that arrive seasonally and can be observed from cage diving vessels deployed specifically for that purpose. Individual sharks at Guadalupe have been identified and catalogued by researchers using the distinctive notching and scarring patterns on their dorsal fins, which are as individually unique as fingerprints. The cataloging effort has built up records on dozens of individual animals over multiple seasons, allowing researchers to track the same individuals returning year after year, to note changes in their condition, and to observe social interactions at the surface. At feeding sites where multiple sharks are present simultaneously, dominance hierarchies have been observed. Larger animals tend to feed before smaller ones. Displacement behaviors, where one shark moves away from a carcass or feeding position as another approaches, have been documented without escalation into direct conflict in most cases. Whether these interactions represent a stable social structure or simply size-based deference in the moment is a question researchers have not fully resolved. The epipelagic zone, the sunlit layer from the surface to around 200 meters, is where white sharks spend the majority of their active time. The prey they depend on most heavily, seals, sea lions, large fish, and other sharks, concentrates in this zone because that is where light penetrates and where the food web is densest. The shark is built for this environment, fast, warm, sensorially equipped, and large enough that very little in the zone presents a threat to an adult animal. Killer whales have been documented killing white sharks at several locations, including off the coast of South Africa and California, by flipping them upside down and inducing tonic immobility. Aside from that documented predation, a fully grown white shark in the sunlit zone has no natural predator. Before we move on, if you're enjoying this analysis, please consider subscribing. It helps the channel tremendously. The next section leaves the open water and moves into the coral reef, the most biodiverse zone in the ocean, where the most dangerous animal is not the largest one present, but one of the smallest, and one that most people would pick up without hesitation. Section 3. The Coral Reef Zone. Cone snail coral reefs cover less than 1% of the ocean floor. The figure is so small, it barely registers against the scale of the ocean, yet those reefs support an estimated 25% of all known marine species. The density of life on a healthy reef is unlike anything else in the sea. Fish in concentrations that make the water look solid, crustaceans working every surface, mollusks packed into crevices, predators threading through all of it in every direction. Hidden in that density, resting on the sand or tucked under rubble during the day and moving at night, is Conus geographus. It is a snail. Its shell is the reason it keeps killing people. The shell of Conus geographus is broadly conical, tapering from a wide body wall to a low spire at one end. The exterior is a creamy off-white, patterned with irregular brown markings that arrange themselves into tent-like shapes across the surface, the pattern that gives the species its common name, the geography cone. It reaches around 15 centimeters in length in adult specimens. On a reef or washed up in the shallows, it looks like a collectible object, which is precisely why the majority of documented human envenomations involve people who picked one up. The shell is attractive in the direct sense of the word. It draws people toward it. The animal inside it responds to being handled. There are around 700 to 900 recognized species of cone snail, all in the genus Conus, distributed through tropical and subtropical marine environments around the world. They are divided broadly by prey type. Some hunt marine worms, some hunt other mollusks, and some hunt fish. The fish hunters are the most dangerous to humans, and Conus geographus is the most dangerous of the fish hunters. Every species in the genus is venomous. The venom delivery system is the same across all of them. A modified tooth, called a radula tooth, hollow, needle-like, loaded with venom, and connected to a muscular proboscis that the animal can extend rapidly in almost any direction from the opening of the shell. The proboscis is not restricted to the opening at the wide end of the shell. It can emerge from what feels like the closed, narrow end. There is no way to hold a live cone snail that places the hand outside the potential reach of that proboscis. People who have learned this fact after picking one up have reported being stung through the palm, through the side of the finger, and through a closed fist. The shell looks inert. The animal inside it is not. The venom of Conus geographus is a cocktail of hundreds of individual peptide compounds called conotoxins, or conopeptides. Unlike the venom of the box jellyfish, which attacks cell membranes broadly, conotoxins are precise. Each peptide targets a specific ion channel or receptor in the nervous system. Some block the sodium channels that allow nerve signals to travel. Others block calcium channels involved in neurotransmitter release. Others target potassium channels. The combined effect of the mixture is a rapid shutdown of neuromuscular signaling. The muscles stop receiving the commands to contract. Paralysis follows, typically beginning locally at the sting site and spreading. In a serious envenomation by Conus geographus, the paralysis reaches the respiratory muscles. The victim stops breathing. Without mechanical ventilation, death follows from respiratory failure. The initial sting is often described by survivors as mild, a sharp prick, sometimes barely noticed in the moment. There is no immediate intensity of pain comparable to the box jellyfish. The venom begins working before the pain is significant enough to prompt alarm. Early symptoms include localized swelling and numbness at the sting site, which can spread over minutes to hours. Vision changes have been reported, including blurring and double vision. Slurred speech, difficulty swallowing, and generalized weakness follow as the neuromuscular blockade deepens. The timeline from sting to respiratory failure varies by the amount of venom injected and the size and health of the victim. But documented cases have progressed from sting to death in under 5 hours, and some reports suggest faster progression. There is no antivenom for any cone snail species. Treatment is entirely supportive. Keep the patient breathing, maintain circulation, wait for the venom to clear the system. Without intensive medical care, the estimated fatality rate for serious Conus geographus envenomation runs at around 65% in older medical literature. That figure reflects cases where full supportive care was not available. The total number of confirmed human deaths attributed to cone snails in the published medical record is approximately 36, with Conus geographus responsible for the majority. That number is certainly an undercount. Cone snail envenomations in remote coastal communities in the Indo-Pacific, where the reefs are and where the shells wash ashore, are not reliably captured in international medical reporting. A fisherman stung on a beach in a rural province who dies before reaching a hospital may not enter the database. Researchers have consistently noted that the actual mortality figure is likely higher than the confirmed total, though how much higher is not possible to determine from available data. The range of Conus geographus runs through the Indo-Pacific reef zone, from the eastern coast of Africa through the Red Sea, across South and Southeast Asia, into the Pacific island chains. The highest density of reef habitat, and therefore the highest concentration of cone snail diversity, falls within the Coral Triangle, a region bounded roughly by the Philippines, Indonesia, Papua New Guinea, Solomon Islands, Timor-Leste, and Malaysia. This region holds more coral species, more fish species, and more cone snail species than any comparable area on Earth. It is also a region with large coastal populations that fish, dive, and walk reef flats as part of daily life. The intersection of that human presence with that cone snail diversity is where the majority of documented stings have occurred. Baldomero Oliveira, a biochemist born in the Philippines who built his research career at the University of Utah, has spent decades studying cone snail venom and is responsible for much of what is currently understood about conotoxin pharmacology. Oliveira grew up near reefs, where cone snails were common. When he established his laboratory, he directed sustained research effort toward characterizing the individual peptide components of cone venom, a project that turned out to be far larger than initially apparent. Each species of cone snail produces its own distinct venom cocktail, and the cocktails do not overlap neatly. The diversity of conotoxin compounds across the genus is extensive enough that researchers have described the cone snails collectively as a vast library of pharmacologically active compounds, each one a precisely engineered key for a specific biological lock. The library is nowhere near fully catalogued. That pharmacological precision is what made cone snail venom interesting to medicine. Ziconotide, a synthetic version of a conotoxin originally isolated from Conus magus, a smaller and less dangerous species, was approved by the United States Food and Drug Administration in 2004 as a treatment for severe chronic pain. It works by blocking a specific calcium channel in spinal cord neurons involved in pain signaling. It is used in patients for whom other pain management approaches have failed, administered directly into the spinal fluid via an implanted pump. The compound is many times more potent than morphine for certain types of pain and does not produce the tolerance and dependence associated with opioid treatment. Ziconotide represents one confirmed medical application. Researchers are actively investigating conotoxins from multiple species for potential applications in pain management, epilepsy, cardiovascular disease, and other conditions. The pharmaceutical interest in the genus has added another layer of motivation to ongoing taxonomy and venom characterization work. A finding published in 2015 by researchers at the University of Utah added an unexpected dimension to the understanding of how Conus geographus hunts. The research found that the species produces a venom component that resembles insulin, the hormone that regulates blood sugar. The hypothesis the researchers proposed is that Conus geographus, when hunting a school of small fish, releases this insulin-like compound into the surrounding water. The fish absorb it through their gills, their blood sugar drops. The resulting hypoglycemia slows them, impairs their ability to swim and respond, and makes them easier for the slow-moving snail to approach and sting with its radula tooth. If the hypothesis is correct, the snail is chemically sedating its prey before the final strike, using a compound that acts on the prey's own metabolic regulation. The research was based on venom analysis and laboratory behavioral observations rather than direct observation of the hunting sequence in the wild, and the authors were careful to describe it as a proposed mechanism rather than a confirmed one. Subsequent work has supported the plausibility of the finding without resolving every aspect of it. The hunting sequence itself, when observed in controlled settings, involves the cone snail extending its siphon to sample chemicals in the water and detect the presence of prey. Once a target is located, the snail extends its proboscis slowly, positioning the radula tooth. The strike is rapid relative to everything else about the animal's movement. The tooth is driven into the prey with enough force to penetrate scales. The venom enters immediately. Fish struck by Conus geographus are immobilized quickly, and the snail then extends its mouth, which can expand dramatically relative to the shell opening, to engulf the prey whole. The snail is capable of consuming fish larger than would seem possible given its shell dimensions, because the oral opening and the tissue around it are highly extensible. The reef itself provides the environmental conditions that make all of this possible. Coral reefs are structurally complex, offering an abundance of crevices, rubble zones, and sandy patches between coral heads where a resting cone snail is almost invisible. The reef's density of fish life gives the geography cone a reliable prey supply. The same structural complexity that makes reefs visually overwhelming to human observers makes them ideal habitat for an ambush predator that relies on concealment and chemical detection rather than speed. The cone snail does not need to be fast. It needs to be patient, chemically sophisticated, and precisely armed. The reef provides the cover. The venom provides everything else. The textile cone, Conus textile, deserves mention alongside the geography cone because it is the second species most frequently implicated in human fatalities. It is a mollusk hunter rather than a fish hunter, with a venom composition tuned accordingly, but the same delivery system applies, and the same danger of handling a live shell exists. The textile cone's shell is among the most sought after by collectors, precisely because its surface pattern, a series of fine overlapping triangles in brown and white, is visually striking. Both the geography cone and the textile cone are found in the same reef habitats across the same broad Indo-Pacific range. A collector working a reef flat who cannot reliably distinguish live shells from empty ones by weight and the feel of the animal inside is handling potential lethality with every pickup. The broader cone snail genus continues to be taxonomically active. New species are described periodically as molecular analysis separates what appeared morphologically similar into genetically distinct lineages. Each newly confirmed species represents a potentially distinct venom profile, meaning the total pharmacological diversity of the genus is not yet known. Researchers working on conotoxin characterization have noted that the pace of new compound identification from cone snails has not slowed despite decades of work. The genus is, in research terms, still being opened. The coral reef zone is the most biologically productive shallow marine environment on Earth, and that productivity means it is also one of the most medically significant. More venomous species are concentrated on reefs than in any comparable zone. Stonefish, the most venomous fish by formal classification, live on reefs. Blue-ringed octopuses, which deliver tetrodotoxin through a bite and are responsible for documented deaths in the Indo-Pacific, live on reefs and in adjacent rock pools. The crown-of-thorns starfish, which has caused severe injuries to divers through its venomous spines, is a reef species. The reef is not uniquely dangerous compared to other zones. It is uniquely dense, and the danger in it is distributed through that density in forms that do not always announce themselves. The geography cone sitting motionless on the sand, patterned like a decorative object, is the clearest example of how completely concealed that danger can be. Before we move on, if you're enjoying this analysis, please consider subscribing. It helps the channel tremendously. The next section moves out of the open reef water and into the brackish zone, where rivers meet the sea, a habitat most people overlook entirely, where the largest living reptile on Earth has been making ocean crossings between islands that researchers tracked only recently. Section 4. The Mangrove and Estuary Zone. Saltwater crocodile researchers at James Cook University fitted satellite transmitters to 27 saltwater crocodiles in northern Queensland and tracked their movements over 12 months. One large male left his river estuary, entered open coastal water, and traveled 590 kilometers in 25 days. He did not swim against the sea, he rode it. The tracking data showed the animal moving with tidal surface currents, drifting when the current ran in his direction, and resting motionless near the bottom when it ran against him. He was using the ocean the way a vessel uses wind, reading the water and timing his movement to it. The finding, published in the Journal of Animal Ecology in 2010, reframed how researchers understood the distribution of Crocodylus porosus across the Indo-Pacific, because an animal capable of covering nearly 600 kilometers on a single ocean passage can colonize islands and coastlines across a range that a purely riverine predator could never reach. Crocodylus porosus is the saltwater crocodile, also called the estuarine crocodile, and it is the largest living reptile on Earth. Confirmed male lengths exceed 6 meters. Large males weigh over 1,000 kilograms, with some specimens in the historical record attributed weights approaching 1,500 kilograms, though the upper end of those historical figures requires caution because early measurements were not always conducted under controlled conditions. Females are considerably smaller, typically reaching around 3 meters. The species holds the laboratory-measured record for bite force among living animals. Researchers at Florida State University, as part of a broader study measuring bite force across crocodilian species, recorded forces from saltwater crocodiles that exceeded those of every other animal tested. The exact figure varies by the size of the individual tested, but the mechanism behind it is the same in every specimen. The jaw-closing musculature is massively developed, occupying a large proportion of the skull volume, while the jaw-opening muscles are comparatively weak. A human hand can hold a crocodile's jaws shut. The same hand cannot survive them opening on it. The mangrove and estuary zone is where Crocodylus porosus is most consistently found. Mangroves are coastal forests that grow where rivers discharge into the sea, in the brackish zone, where fresh and salt water mix, and where the salinity fluctuates with the tides. Mangrove trees grow on root systems that arch above the waterline, creating a dense tangle of aerial roots at the waterline, called pneumatophores, and prop roots that trap sediment and build the coastline outward over time. The water beneath the canopy is dark, slow-moving, and visually opaque. The root tangle breaks the surface into small channels and pockets. Navigation through it is slow and requires attention. For a large ambush predator, it is close to ideal habitat. Abundant cover, reliable water, proximity to prey moving between the river and the sea, and a structure that makes detection of the predator by that prey very difficult. The saltwater crocodile's ability to use this habitat is supported by a sensory system significantly more sophisticated than its prehistoric appearance might suggest. Along the jaw and across the body, scales are small, dark, dome-shaped structures called dome pressure receptors. These structures, found in all crocodilians, but particularly well developed in the saltwater species, detect pressure changes and vibrations in the surrounding water with a sensitivity that has been compared in research literature to the lateral line system of fish. A crocodile lying motionless on the floor of a mangrove channel can detect the pressure wave generated by an animal moving through the water above it. It can detect the surface disturbance caused by a raindrop. The system allows the animal to track the position and movement of prey in conditions where vision is useless, which describes most of the water column in a mangrove environment. The crocodile does not need to see what it is hunting. It needs to feel the water move. The attack itself, when it comes, follows a pattern documented across decades of wildlife observation and attack incident reports. The crocodile approaches from below or from the side in a lunge that covers the final distance in a fraction of a second. The initial bite is directed at securing the prey, often targeting the head, neck, or a limb. Once the bite is established, the animal either holds and drowns the prey by remaining still underwater or performs what is called a death roll, a rapid rotation of the entire body around the long axis. The death roll is not simply aggression. It functions to disorient prey, to overcome the resistance of larger animals, and to tear limbs and chunks of flesh free from prey too large to swallow whole. Crocodilians lack the ability to chew or manipulate food inside their mouths. They swallow pieces. The death roll is the mechanism by which large prey is reduced to pieces of a swallowable size. It has been observed in captive animals in controlled conditions and is documented extensively in wildlife camera footage from attack sites. The lingual salt glands on the tongue of Crocodylus porosus are what allow the species to tolerate prolonged exposure to seawater. These glands actively excrete salt, allowing the animal to regulate its internal salt balance while traveling through ocean water. Freshwater crocodiles and some other crocodilian species lack fully functional salt glands of this type. The saltwater crocodile's glands are one of the biological features that separate it from purely freshwater relatives and make the documented ocean crossings physiologically possible. Combined with the current surfing behavior identified by the James Cook University tracking study, the salt glands mean the animal has both the physiological capacity and the behavioral strategy to use the open ocean as a corridor between river systems. The range of Crocodylus porosus extends from the eastern coast of India and Sri Lanka through Bangladesh, Myanmar, Thailand, Malaysia, Indonesia, the Philippines, Papua New Guinea, and the Solomon Islands, down to the northern coast of Australia. Australia holds the most extensively studied and best monitored population, concentrated in the Northern Territory and northern Queensland. In the mid-20th century, the Australian population had been reduced to a fraction of its historical numbers by hunting for the leather trade. Saltwater crocodile skins were commercially valuable, and the animals were hunted intensively across northern Australia from roughly the 1940s through the 1960s. By the time legal protection was introduced, in the Northern Territory in 1971 and in Queensland in 1974, population numbers had collapsed in many areas. The recovery since protection has been substantial. Current population estimates for the Northern Territory alone run above 100,000 animals, a figure that represents a recovery over 5 decades that wildlife managers have described as one of the more successful large predator conservation outcomes in Australia. That recovery has had direct consequences for human safety in northern Australia, because more crocodiles across a wider range means more encounters between the animals and people using the same waterways. Fatal attacks in the Northern Territory and Queensland are documented annually, averaging roughly 1 to 2 confirmed deaths per year in Australia in recent decades, though the figure varies. Non-fatal attacks occur at higher frequency and are tracked through a database maintained by researchers, including Adam Britton, a crocodile biologist who has contributed substantially to the published literature on the species. Britton's attack database, compiled over years from hospital records, police reports, and media documentation, represents one of the more comprehensive records of human crocodile encounter outcomes available for any crocodilian species. The documented attack patterns show a concentration in predictable circumstances, people wading, swimming, or crouching at the water's edge in areas of known crocodile habitat, often at dawn or dusk when the animals are most active at the surface. A significant proportion of attacks in the Australian record involve people who were aware of the crocodile warning signs in the area and entered the water anyway, or who were engaged in activities, cleaning fish at the water's edge, setting nets, swimming in known crocodile territory, that placed them in the water at the times and locations where encounters are most likely. This is not a criticism of the victims. It reflects the difficulty of maintaining consistent caution in environments that people use regularly and familiarly, where the risk feels abstract until it is not. Outside Australia, documentation is less systematic, but the human toll is considered higher in absolute terms. Papua New Guinea and Indonesia both have large coastal populations living in close proximity to mangrove and estuary habitat, with limited access to emergency medical care in many areas, and with crocodile populations that have faced varying levels of hunting pressure over the decades. Researchers working in those regions have consistently noted that the true frequency of fatal attacks is likely substantially underreported relative to the Australian figures. The World War II Battle of Ramree Island, off the coast of what is now Myanmar, produced one of the most cited and most disputed claims in the documented history of saltwater crocodile attacks. In February 1945, British forces moved to encircle a Japanese garrison on the island. A significant number of Japanese soldiers, estimates vary widely in the historical record, retreated into the mangrove swamps rather than surrender. A British naturalist named Bruce Stanley Wright, who was present during the operation, later wrote an account describing the sounds coming from the mangroves during the night, gunfire, animal sounds, and the screams of men, and attributed hundreds of deaths to crocodile predation. The account was picked up by the Guinness World Records and repeated widely as a documented fact. Military historians have since raised serious questions about the figure. Japanese records, casualty accounting, and the logistics of the retreat do not support a death toll in the hundreds from crocodile attacks specifically. The true number of soldiers who died in the swamps from all causes combined, including wounds, disease, and exposure, as well as any crocodile predation that did occur, is not established with confidence. The incident is documented as a real event. The specific claim of mass crocodile predation at the scale described by Wright is disputed and should be treated as such. The female saltwater crocodile builds a nest of vegetation and soil at the water's edge, typically in a location sheltered from flooding but close enough to water for the female to monitor from the surface nearby. Clutch sizes average between 40 and 60 eggs. Incubation lasts around 90 days, and the temperature of the nest during incubation determines the sex of the hatchlings, a system called temperature-dependent sex determination shared with many other reptiles. The female guards the nest aggressively and responds to the vocalizations of hatching young by opening the nest and sometimes carrying hatchlings to the water in her mouth. Despite this parental investment, hatchling mortality is extremely high. Most juveniles are taken by predators, including monitor lizards, large fish, birds, and other crocodiles, before reaching a size at which they can defend themselves effectively. Of the 40 to 60 eggs in a typical nest, the fraction that survive to adulthood is small. Adults, once past a threshold size, have no natural predators within their range. A fully grown saltwater crocodile in a mangrove estuary occupies the apex of the food web absolutely. There is no other animal in the zone that presents a threat to it. The combination of size, bite force, sensory capability, ambush behavior, and tolerance for both fresh and saltwater places the species in a category that has very few parallels in any environment. The mangrove zone it inhabits is one of the least visited and least understood coastal environments, a fact that has probably contributed to the species being underrepresented in public awareness relative to its biological significance. It is not a reef animal or an open ocean animal. It lives in the transition zone, the murky edge where the river becomes the sea, and it has been doing so, in a body plan largely unchanged from its ancestors, for a very long time. Before we move on, if you're enjoying this analysis, please consider subscribing. It helps the channel tremendously. The next section leaves the coast entirely and moves into the open ocean, where an animal that is not one animal, but 4 distinct organisms fused into a single body drifts on the surface currents, trailing tentacles 30 meters long through the water below. Section 5. The Open Ocean. Portuguese man-o'-war Physalia physalis is a colony of 4 distinct types of organism, each type called a zooid, each zooid a separate living entity with its own specialized function. none of them capable of independent survival, all of them operating together as a unit, so integrated that the colony functions as a single predator moving through the open ocean. The way it moves, the way it feeds, the way it stings, and the way detached fragments of it continue operating hours after separation from the main colony all follow directly from that colonial architecture. The Portuguese man-o'-war takes its common name from the Portuguese sailing warships of the 15th and 16th centuries. Seen from a distance at sea level, the float riding the surface with its crest raised, the resemblance to a warship under sail was apparently close enough to stick. The float itself, called the pneumatophore, is the most visible part of the colony. It is a gas-filled bladder reaching up to 30 centimeters in length, translucent, and tinted in shades of blue, pink, and violet, with a stiffened crest running along the top that functions as a sail. The gas mixture inside is primarily carbon monoxide, produced by the colony through a biological process rather than extracted from the surrounding air. The float sits at the surface. Everything else hangs below it. Below the pneumatophore, the other three zooid types carry out the colony's biological work. The dactylozooids are the tentacles, long contractile strands covered in nematocysts, the same pressurized sting capsules found in true jellyfish. They are responsible for capturing prey. The gastrozoids handle digestion, absorbing nutrients from captured prey and distributing them through the colony. The gonozoids manage reproduction. Each zooid type is structurally and functionally specialized to a degree that it cannot perform the functions of the others. The tentacle zooids cannot digest. The digestive zoozoids cannot sting. The colony is a division of biological labor taken to an extreme that has no equivalent among solitary animals. The tentacles of Physalia physalis average around 10 meters in the water during normal trailing, but they are highly contractile and can extend considerably further. Confirmed measurements of tentacle length from large specimens have reached 30 meters. The tentacles drift below and behind the float, carried by the same current that carries the colony, spreading across a wide area of water in a net-like arrangement that the colony makes no active effort to position. It does not maneuver its tentacles toward prey. It drifts through the water and the tentacles encounter prey by contact. When contact occurs, the nematocysts fire, injecting venom into whatever triggered them. Small fish, crustaceans, and other zooplankton are the primary prey. The tentacles then contract, drawing the immobilized prey up toward the gastrozoids beneath the float. The colony cannot swim. The pneumatophore cannot be deflated or submerged. Physalia physalis moves entirely at the mercy of wind and surface current, with the sail crest providing the only mechanism of directional influence. Research has found that the orientation of that crest is not uniform across the population. Roughly half of all man-o'-war colonies in a given area are oriented with the crest angled to sail to the right of the wind direction, and roughly half are oriented to sail to the left. The proportion has been documented in multiple studies as approximately equal in most populations. The leading hypothesis is that this bilateral distribution spreads colonies across different wind-driven pathways, reducing the likelihood that the entire population concentrates in one location and exhausts the prey there. Whether this is an adaptation that evolved through selection or an incidental consequence of developmental variation is a question that has not been fully resolved, but the equal distribution across sail orientations is well documented and consistent across populations studied in different ocean basins. The venom of Physalia physalis is complex and has been studied extensively, though characterizing it precisely is complicated by the fact that venom composition varies somewhat between specimens and between populations in different geographic regions. The core toxic effects involve pain at the contact site, which is immediate and intense, followed by inflammation and a characteristic whip-like welt that traces the path of the tentacle across the skin. Systemic effects in more serious cases include fever, muscle cramps, nausea, and in rare documented instances, cardiac and respiratory complications. Deaths attributed directly to man-o'-war venom are rare in the medical literature, but the circumstances of some fatal encounters are more complex. A swimmer, stung by multiple long tentacles in open water, experiencing sudden severe pain, may panic, inhale water, and drown before reaching safety. The venom is the initiating cause, the drowning is the mechanism. In those cases, the fatality may be recorded differently depending on the jurisdiction and the thoroughness of the post-mortem investigation. The first aid question for Physalia physalis stings has been substantially revised in recent years. For decades, the recommended approach for jellyfish and man-o-war stings broadly tracked together, with various treatments including cold packs, alcohol, and other substances recommended at different times by different sources. Research published in medical toxicology literature around 2017 compared treatment approaches directly and found that immersion of the stung area in hot water, around 45 degrees Celsius, significantly reduced pain compared to cold treatment and compared to other commonly recommended first aid measures. The research also found that vinegar, which is effective for box jellyfish stings because it deactivates unfired nematocysts, should not be used for Physalia physalis stings. The nematocyst type in Physalia physalis responds differently to vinegar, and application may cause unfired nematocysts still adhering to the skin to discharge. Rinsing with seawater rather than fresh water is the current recommended approach for initial removal of tentacle material, as fresh water can also trigger remaining nematocysts. The revision of these recommendations based on direct experimental evidence has taken time to work its way into public awareness, and older advice still circulates widely. Detached tentacles present a hazard that extends beyond the colony itself. Physalia physalis strands on beaches in large numbers when onshore winds push surface water toward the coast. The float dries and deflates relatively quickly in air. The tentacles do not. They lose moisture but retain functional nematocysts for hours after stranding, and in some documented conditions considerably longer. A tentacle fragment lying on wet sand, to the eye indistinguishable from a piece of clear plastic or a strand of seaweed, retains the ability to sting anyone who steps on it or picks it up. The majority of man-o-war stings in heavily visited beach areas occur not in the water from live colonies, but on or near the shoreline from stranded material. In Australia, where the Indo-Pacific species Physalia utriculus, known locally as the bluebottle, strands in enormous numbers on East Coast beaches after northerly winds, the annual sting count runs into the tens of thousands. Life-Saving Australia and the organizations tracking beach safety incidents describe the bluebottle as responsible for more stings per year than any other marine animal in Australian waters. The relationship between Physalia physalis and other species in the open ocean is not purely predatory in one direction. Several animals use the man-o-war as habitat, as food, or both. The man-of-war fish, Nomius granovii, lives among the tentacles of Physalia physalis, apparently resistant to the venom or capable of avoiding sting contact through precise movement, feeding on the tentacle tissue itself and on small organisms attracted to the colony. Young man-of-war fish have been observed darting in and out of the tentacle mass without apparent harm, though they are not fully immune and documented sting injuries to them have been observed in some studies. The loggerhead sea turtle feeds on Physalia physalis and appears fully immune to the venom, consuming float, tentacles, and all without visible ill effect. Loggerhead turtles have been documented with facial scarring from repeated man-o-war encounters that suggests regular consumption of the colonies throughout the turtle's lifetime. The blue sea slug, Glaucus atlanticus, has one of the stranger relationships with Physalia physalis in the open ocean. Glaucus atlanticus is a small nudibranch, rarely more than 3 centimeters long, that floats at the surface upside down, held up by a bubble of air it stores in its stomach. It feeds on Physalia physalis and on other surface-floating organisms, including the by-the-wind sailor, Velella velella, a smaller colonial organism often confused with the man-o-war, but structurally quite different. When Glaucus atlanticus consumes Physalia physalis nematocysts, it does not digest them. It sequesters them, moving them through its own tissue into structures at the tips of its appendages, where they are stored functional and ready to discharge. The nudibranch thereby acquires the sting of its prey without producing venom of its own, a process called kleptocnidae. Because Glaucus atlanticus concentrates nematocysts from multiple feeding events into its appendage tips, its sting can be more potent than that of the original Physalia physalis tissue it consumed, a counterintuitive outcome that has surprised people who have handled the small, visually striking slug under the assumption that something so small and colorful encountered on a beach is harmless. The siphonophore group to which Physalia physalis belongs includes some of the structurally largest organisms ever recorded. Prior dubia, a deep-water siphonophore of the open ocean, has been measured at lengths exceeding 40 meters, considerably longer than the largest blue whale. That length is achieved through the same colonial zooid structure as Physalia physalis, but Praia dubia is a swimmer rather than a surface drifter, living in the deeper mesopelagic water column and hunting in the dark. It shares with Physalia physalis the fundamental biological architecture of a colonial organism operating as a single predatory unit, a body plan that has produced some of the most successful predators in the open ocean across hundreds of millions of years of evolutionary history. The aggregations Physalia physalis forms when wind and current concentrate large numbers of colonies together are sometimes described as armadas in both popular and scientific literature. Observers have documented patches of ocean surface where colonies stretch in every direction for hundreds of meters. The blue floats visible at intervals across the water, the tentacles below connecting into an overlapping mesh through the upper water column. For a school of small fish swimming through such an aggregation, the encounter would be with a distributed predator covering an area of ocean that no single animal could manage. The colonial architecture of Physalia physalis scales in a way that individual body plans cannot. One colony trailing 30-meter tentacles covers a significant volume of water. Hundreds of colonies in proximity cover a volume with few equivalents in open ocean predation. The open ocean, the pelagic zone beyond the continental shelves and away from any coast, is the largest habitat on Earth by volume. It is also among the least understood because it is vast, featureless to the eye, and difficult to sample systematically. Physalia physalis is one of the most visible occupants of its surface layer, precisely because it cannot submerge. It is carried where the wind and current take it, visible when the sun hits the float at the right angle, concentrated by coastal waters when onshore winds push it ashore, and largely invisible at all other times to anything that is not in the water with it. The open ocean conceals almost everything that lives in it. Physalia physalis is one of the rare exceptions, floating at the boundary between sea and air, with nowhere else to go. Before we move on, if you're enjoying this analysis, please consider subscribing. It helps the channel tremendously. The next section descends below the sunlit surface entirely, into the permanent twilight between 200 and 1,000 meters, where an animal considered mythological by Western science for most of recorded history was finally photographed alive in its natural habitat in 2004. Section 6. The Twilight Zone. Giant Squid. On September 30th, 2004, an automated camera rig lowered to 900 meters depth near the Ogasawara Islands in the North Pacific triggered on something large, moving toward the bait attached to the line above it. The rig had been deployed by researchers Tsunemi Kubodera of the National Science Museum of Japan and Kyoichi Mori of the Ogasawara Whale Watching Association. They had been attempting versions of this deployment repeatedly over several years without success. On this occasion, the camera fired 556 frames over a period of several hours. When the rig was retrieved and the images were developed, they showed Architeuthis dux, the giant squid, alive and moving in its natural habitat, for the first time in the history of science. One of the images showed the animal with a tentacle wrapped around the bait line. In a subsequent frame, the squid had pulled free, leaving behind a tentacle tip that had torn off during the struggle. The torn tip, recovered with the rig, measured 5.5 meters in length. The mesopelagic zone runs from 200 to approximately 1,000 meters below the surface. Light from the surface penetrates the upper boundary in diminishing quantities and is effectively absent before the lower boundary is reached. The zone is sometimes called the Twilight Zone, a name that describes the quality of faint, diffuse illumination that characterizes its upper reaches and fades progressively to nothing as depth increases. Pressure at 500 meters is approximately 50 times the pressure at the surface. Temperature drops sharply below the warm, mixed layer of the surface ocean into a cold, stable band that changes little with further depth increases within the zone. Food is scarce compared to the sunlit zone above. What arrives comes primarily as marine snow, the continuous slow fall of dead organic particles, fragments of organisms, and waste material drifting down from above and through the bodies of animals that die in shallower water and sink before being consumed. Animals in the mesopelagic zone are adapted to cold, pressure, scarcity, and diminishing light. and a significant proportion of them produce their own bioluminescence to navigate and hunt within it. Architeuthis dux lives across this zone and almost certainly below it, though the full depth range is not precisely established from direct observation. It is the longest invertebrate confirmed by science to exist. Females grow considerably larger than males, a pattern of sexual size dimorphism that runs through cephalopods broadly but reaches its most extreme expression in the giant squid. The mantle, the main torpedo-shaped body section, not including the arms and tentacles, reaches confirmed lengths of around 2 meters in large females, with some specimens measured beyond that. Adding the 2 long feeding tentacles that extend well past the 8 shorter arms, total length estimates for large specimens appear in the scientific literature in the range of 12 to 13 meters, though the methodology behind the largest published figures has been examined critically by some researchers. A giant squid is highly contractile, capable of shortening and extending its arms and tentacles dramatically, and measuring a recently dead or damaged specimen that may have been torn or compressed during trawling introduces significant uncertainty. What is consistently supported across the literature is that females are substantially larger than males, that the animal is the longest invertebrate on Earth, and that individual specimens vary considerably in size in ways that make single maximum length claims difficult to evaluate without knowing the measurement conditions. The eyes of Architeuthis dux reach 30 centimeters in diameter in large specimens. This is the largest eye of any living animal, and by a margin that separates it clearly from the next largest, which belong to the colossal squid Mesonychoteuthis hamiltoni, a distinct species from Antarctic waters. Eyes of 30 centimeters in an environment of total darkness appear paradoxical until the predator that hunts giant squid is considered. Sperm whales descend into the mesopelagic zone to feed, and Architeuthis dux is among their prey. Sperm whales are large, warm, and moving, and as a large warm animal moves through dark water, it displaces bioluminescent organisms in the water column around it, generating a faint moving glow from the disturbed plankton. The leading hypothesis for the evolution of the giant squid's enormous eyes, developed and modeled by researchers including Dan Erik Nilsson at Lund University, is that the eye evolved specifically to detect this bioluminescent disturbance at a distance sufficient to give the squid time to flee. A smaller eye would gather insufficient light to resolve a moving whale against the background darkness at any useful distance. The 30-centimeter eye provides enough light-gathering area to detect the faint glow of a descending whale before the whale is within striking range. The optical modeling supporting this hypothesis has been published and reviewed, and while it remains a hypothesis rather than a confirmed fact, it is the most developed and evidence-supported explanation for why an invertebrate living in darkness needs an eye of that size. The evidence for the predatory relationship between sperm whales and giant squid comes from 2 sources, and both have been available to researchers for well over a century. The first is the contents of sperm whale stomachs. Sperm whales feed extensively on squid across a range of sizes and species, and their stomachs contain indigestible squid beaks in large numbers because the hard chitin of the beak resists the digestive processes that break down everything else. Giant squid beaks have been recovered from sperm whale stomachs and intestines, identified by size, against the relationship between beak dimensions and body size established from measured specimens. The accumulation of indigestible squid beaks in the lower digestive tract of sperm whales is one of the processes thought to contribute to the formation of ambergris, a dense, waxy substance that forms around concentrations of indigestible material in the sperm whale gut, hardens over time, and was historically among the most valuable commodities in the perfume industry, used as a fixative that extended the persistence of scent compounds. The second source of evidence is the skin of sperm whales. Circular scars, matching the dimensions of giant squid sucker rings, have been documented in photographs of living and dead sperm whales. Giant squid suckers are ringed with hard, tooth-like serrations, and the marks these leave on whale skin are distinctive. Researchers have used the diameter of these scar rings and the known relationship between sucker diameter and total body length to estimate the size of squid involved in specific encounters. The scars are found on animals that survived the encounter, indicating that at least some interactions between sperm whales and giant squid involve significant physical resistance from the squid before or during capture. The internal anatomy of Architeuthis dux includes several features that reflect its adaptation to the mesopelagic environment. The tissues of the giant squid are permeated with a solution of ammonium chloride, which is less dense than seawater, and provides the animal with near-neutral buoyancy throughout the water column, without the need for a gas-filled swim bladder of the kind fish use. The trade-off is that the tissue is rendered chemically unpleasant to eat, with a strong ammoniacal flavor that has been described by researchers who have sampled it as ranging from intensely bitter to frankly inedible. The giant squid is consequently not commercially fished for human consumption despite its size, and encounters between giant squid and fishing vessels are accidental bycatch rather than targeted harvest. The ammonium chloride also means that specimens recovered for museum collection must be handled carefully, as the compound continues to leach from the tissue during preservation and handling. The arms and tentacles of Architeuthis dux are equipped with suckers bearing serrated rings, and the 2 long feeding tentacles, which are considerably thinner than the 8 shorter arms, terminate in flattened clubs with a higher density of suckers. The tentacles are the primary prey capture tools. In squid generally, tentacle extension during a strike is among the fastest predatory movements documented in marine invertebrates, achieved through a combination of muscular pressure and hydrostatic force. The specific mechanics of the giant squid's tentacle strike have not been filmed in sufficient detail in natural conditions to characterize precisely, but the body plan is fundamentally the same as in smaller squid species, where high-speed filming has been used to analyze the strike. After tentacle contact immobilizes or secures the prey, the shorter arms close around it, and the prey is drawn toward the beak. The beak is hard, curved, and powerful enough to cut through the bodies of fish, considerably larger than would seem manageable, given the soft-bodied nature of the animal holding it. The radula, a rasping structure common to mollusks, breaks down the prey further before digestion. The Kraken of Norse legend drew directly from real biological encounters, even if the legend itself elaborated far beyond them. The earliest systematic written account appears in Erik Pontoppidan's Natural History of Norway, published in 1752, which described a creature of such size that sailors mistook its surfacing back for an island. The creature of the legend was associated with whirlpools generated by its descent and with catastrophic encounters at sea. The biological basis behind the Scandinavian accounts was almost certainly encounters with giant squid at the surface, possibly disoriented or dying animals, combined with the evidence of enormous squid recovered from the stomachs of stranded sperm whales. Aristotle described what may have been a large squid in his Historia Animalium, using the term teuthus to distinguish a large species from smaller squid, though whether the animal he was describing was Architeuthis or simply a large common squid cannot be resolved from the text. Pliny the Elder wrote of enormous squid encountered near the coast of Spain. Herman Melville, in Moby Dick, described an encounter in which the crew of the Pequod sighted a vast pale mass at the sea surface that they initially mistook for the white whale, before recognizing it as a squid of extraordinary size. Whether Melville was drawing on a specific account he had encountered or constructing the passage from the existing legend is not established. The first physical specimens of Architeuthis dux that could be examined by Western science arrived in the 1870s and 1880s, primarily from Newfoundland. In October 1873, a fisherman named Theophilus Picot and his son encountered a large mass of floating matter off the coast of Conception Bay and rowed toward it. When they poked it with a hook, the animal responded. It seized their small dory with its tentacles. Picot's son, described in the accounts of the incident as 12 years old, cut through one of the tentacles with a hatchet. The squid released the boat and sank. The severed tentacle, reportedly around 5.5 meters long, was brought ashore. A local minister and naturalist named Moses Harvey heard of the incident, acquired the tentacle, and began corresponding with scientists in the United States and Britain. Harvey subsequently acquired a nearly complete specimen that had been caught in a herring net. He draped the mantle over a curtain rod in his bathroom, arranged the arms around it, and photographed it. The photograph, sent to researchers including Addison Emery Verrill at Yale University, provided the first widely distributed physical documentation of the animal. Verrill used Harvey's specimens and photographs as the basis for a series of papers formally describing the giant squid and establishing the scientific literature that has built around it since. The taxonomy of the giant squid accumulated considerable complexity through the 19th and early 20th centuries because each new specimen that came to light was often described as a new species, producing a proliferation of species names within the genus Architeuthis. By the late 20th century, the literature contained references to as many as 21 named species. A study published in 2013, led by Inga Winkelmann and conducted in collaboration with researchers from multiple institutions, applied molecular analysis to tissue samples from 21 specimens collected from locations across the Atlantic, Pacific, and Southern Oceans. The analysis found no significant genetic differentiation between specimens from different ocean basins. The conclusion was that Architeuthis dux is a single globally distributed species, with the variation in appearance between specimens reflecting individual variation, sex, age, and the condition of the specimen rather than true species differences. The finding collapsed the list of named species into one and implied that the giant squid moves through ocean basins in ways that maintain genetic connectivity across its entire range. Though how that connectivity is maintained in an animal that spends its life at depth is not fully understood. 8 years after the 2004 still photographs, Kubodera returned to the Ogasawara Islands with a substantially different approach and a larger team. Working with NHK, the Japanese public broadcasting organization, and the Discovery Channel, he used a specially designed submersible equipped with dim red illumination rather than white light. The reasoning behind the red light was specific. Most deep-sea animals lack photoreceptors sensitive to red wavelengths, making red light functionally invisible to them in a way that white light is not. A white-lit submersible announces its presence to the animals around it. A red-lit submersible may approach without triggering avoidance behavior. The submersible descended to over 900 meters in the waters near the Ogasawara Islands. Kubodera and his colleagues filmed a living giant squid moving through the water column, believed to be the first video footage of Architeuthis dux in its natural environment. The animal in the footage appeared to be approximately 3 meters in mantle length, silver-colored in the red-shifted artificial light, moving by jet propulsion, and showed no apparent response to the submersible's presence until the vessel came quite close. The footage was broadcast as part of documentary productions in Japan and internationally in 2013, and has been widely referenced in subsequent scientific and popular accounts of the species. The colossal squid, Mesonychiteuthis hamiltoni, occupies a different part of the world's ocean and a different ecological niche, but is frequently discussed alongside Architeuthis because the two animals are commonly conflated in popular accounts. The colossal squid is found in Antarctic and subantarctic waters, is heavier-bodied than the giant squid despite potentially similar total lengths, and bears rotating hooks on its tentacle clubs rather than the toothed sucker rings of Architeuthis. The largest confirmed specimen was caught accidentally by the New Zealand fishing vessel San Aspiring in Antarctic waters in 2007. It weighed 495 kilograms and measured approximately 4.5 meters in mantle length. It was frozen aboard the vessel and transported to New Zealand, where it was examined by scientists at the Museum of New Zealand, Te Papa, Tongarewa. The examination took place over several days as the specimen was carefully thawed, and portions of it were broadcast publicly as a live event. The process produced detailed measurements, tissue samples, and close examination of the beak and eye structures, including an eye that measured around 27 centimeters in diameter, only slightly smaller than the largest documented giant squid eyes. The mesopelagic zone that Architeuthis dux inhabits is one of the largest and least understood habitats on Earth by volume. Biomass estimates based on acoustic surveys suggest the zone contains more animal life than all shallower oceanic zones combined, most of it in the form of small fish, crustaceans, and gelatinous organisms that participate in the daily vertical migration, ascending toward the surface each night to feed in darkness and descending before dawn. That migration, repeated every 24 hours by an enormous mass of organisms, represents one of the largest animal movements on Earth, measured by total biomass. The giant squid does not appear to participate in this daily migration in the way smaller mesopelagic animals do. It lives at depth more consistently, connected to the food web above, through the prey that moves through its zone during those migrations, and through the sperm whales that descend from above to hunt within it. Understanding Architeuthis dux with any completeness requires understanding the zone it lives in, and that zone remains, despite recent advances in deep-sea research technology, among the most poorly sampled environments on the planet. Before we move on, if you're enjoying this analysis, please consider subscribing. It helps the channel tremendously. The next section descends below the mesopelagic entirely into the permanent darkness of the midnight zone, where a fish with a bioluminescent lure has solved the problem of finding a mate in the deep ocean through a reproductive strategy that has no equivalent anywhere else in the vertebrate world. Section 7. The Midnight Zone. Anglerfish. Below 1,000 meters, the last trace of sunlight is gone. The bathypelagic zone runs from that depth to around 4,000 meters and is permanent, absolute darkness. Temperature sits near freezing across the entire band. Pressure at 2,000 meters is approximately 200 times the pressure at the surface. Food arrives from above as marine snow, the slow, continuous drift of dead organic material falling from the zones above, and through the bodies of animals that die in shallower water and sink. The deep-sea anglerfish has adapted to this environment by becoming one of the most biologically extreme vertebrates ever documented, with a reproductive strategy that has no equivalent in any other group of vertebrates on Earth. There are more than 160 described species within the suborder Ceracioidea, the group collectively called deep-sea anglerfishes, distributed across every ocean basin. Body plans vary considerably between families within the group, but the defining feature of the female is consistent. A modified dorsal spine called the elysium extends forward from the top of the head and terminates in a fleshy tip called the esker. The esker glows. In the absolute darkness of the bathypelagic zone, the female anglerfish hangs in the water with a point of bioluminescent light positioned in front of her face, and she waits for something to move toward it. The light is produced by bacteria, not by the fish itself. The esker is colonized by bioluminescent bacteria that produce light through a chemical reaction involving the compounds luciferin and luciferase, the same biochemical mechanism responsible for bioluminescence in fireflies and in the many other light-producing organisms distributed through the deep ocean. The bacteria live inside the esker tissue in dense concentrations. The fish provides them with a sheltered environment and nutrients delivered through blood vessels running into the esker. The bacteria provide the light. Neither party produces the outcome independently. How the bacteria colonize the esker as the juvenile female descends from shallower water into the bathypelagic zone is not fully resolved. Research has identified specific bacterial lineages associated with anglerfish eskers, and work continues on the mechanism by which those bacteria find and establish themselves in the esker tissue of a descending juvenile. The relationship is more complex than a simple environmental pickup, and the details of how it initiates and stabilizes are still being characterized. The esker does not produce a steady, constant glow in most species. Many ceratioid anglerfishes are capable of modulating the light output, generating pulses, varying the intensity, and in some cases, producing patterns. The elysium is flexible and can be moved, allowing the female to sway the glowing tip through arcs and motions that in some species closely resemble the movement of a small crustacean or bioluminescent invertebrate. For an animal living in total darkness, where any light source represents a potential food opportunity, following that light toward its apparent source is a behavior selected for across generations. The anglerfish exploits that selection by mimicking the thing other animals are already inclined to approach. Animals drawn toward the esker move into range of the mouth. The mouth of a female ceratioid anglerfish is large relative to the rest of the body, often nearly as wide as the body itself at full gape. The teeth are long, curved inward toward the throat, and in several species are transparent enough to be nearly invisible during examination. An animal that has entered within range of those teeth cannot easily reverse direction. The inward curve prevents retreat. The stomach of the anglerfish is highly distensible, capable of expanding substantially beyond the resting volume of the body. Prey items approaching or exceeding the length of the fish itself have been recovered from stomachs during examination of trawl-caught specimens. This capacity is a direct adaptation to scarcity. In an environment where food is unpredictable and intervals between meals may be extended over days or weeks, the ability to consume and store very large prey items when they are available, provides a meaningful survival advantage. The female anglerfish is the large, formidable, predatory animal that most depictions of the species focus on. For most of the history of scientific work on the group, the male was essentially unknown, and understanding why took several decades of confusion. Female ceratioid anglerfishes were the first specimens collected and formally described, beginning in the late 19th and early 20th centuries. They were recovered in deep trawl nets, preserved, and sent to natural history institutions for examination. The males were not found alongside them, and when small, structurally unusual organisms were eventually found attached to female specimens, they were initially classified as parasites or as juveniles of unrelated species. The first correct identification of a parasitic male ceratioid anglerfish was made by Charles Tate Regan at the Natural History Museum in London in 1922. Regan examined a specimen of Ceratias holboeli, a female with a small worm-like organism fused to her underside, and identified it correctly as a male of the same species. He described the relationship as sexual parasitism, a term that has persisted in the literature. The publication of that identification opened a period of reassessment of previously collected small organisms that had been catalogued separately, many of which were eventually reclassified as males of species whose females were already described. The male ceratioid anglerfish is dramatically smaller than the female. Across the group, the size disparity is the most extreme documented between males and females of the same species among vertebrates. In some species, females grow to lengths exceeding a meter, while males of the same species reach under 2 centimeters. The male hatches from a pelagic egg in the upper water column and develops as a free-swimming larva with functional, well-developed eyes and a highly developed olfactory system. He does not feed as an adult. His digestive system begins degenerating before or shortly after he achieves sexual maturity, meaning he has a limited window in which to locate a female before he starves. The pressure this places on the efficiency of that search is reflected in his sensory apparatus. The olfactory organs of male ceratioid anglerfishes are proportionally very large, specialized for detecting the specific pheromone compounds released by females of the same species in the water around them. He follows that chemical gradient downward, through the water column, into the darkness. When the male finds a female, he bites into her skin. Enzymes at the contact site begin dissolving the tissue of both animals at the junction point. The surfaces fuse. Blood vessels from the male connect with blood vessels from the female. Circulation becomes shared. The connection is not external attachment in the way a parasite might attach to a host surface. It is internal, at the level of joined vasculature, with blood flowing between two originally separate individuals through fused tissue. After this fusion is complete, the male's body undergoes a systematic reduction. His eyes degenerate and cease to function. His olfactory organs, no longer needed, degenerate. His internal organs, with the exception of the testes, cease functioning and are progressively reabsorbed into the shared tissue. What remains attached to the female is a reproductive structure supplied by her circulatory system, receiving nutrients through her blood and capable of releasing sperm when her eggs are ready to be fertilized. The male does not feed independently again. He does not navigate independently again. He exists, for the remainder of the female's life, as a component of her body. A single female can carry multiple males simultaneously. Each one found her independently, located her through the same pheromone gradient, and fused at a different location on her body. Specimens with as many as 8 attached males have been documented in the scientific literature. Each of those males is a separate individual, carrying different genetic material, fused permanently to the same female, all of them supplied by her single circulatory system. The arrangement maximizes the female's access to sperm across multiple reproductive events. In the deep ocean, where population densities are low and encounters between individuals of the same species are infrequent, carrying multiple males eliminates the need to locate a mate at the moment of each spawning event. The immunological dimension of this arrangement occupied researchers for decades because it appeared to contradict fundamental principles of vertebrate immunology. The adaptive immune system of vertebrates identifies foreign tissue through molecular markers on cell surfaces and mounts a rejection response against it. Organ and tissue transplants between genetically distinct individuals of the same species require immunosuppressive drugs to prevent the host's immune system from destroying the transplanted tissue. Two unrelated individuals sharing a circulatory system indefinitely should, under normal vertebrate immunology, produce mutual rejection. Neither animal should be able to tolerate the other's tissue. Research published in 2020, led by scientists at the Max Planck Institute for Immunobiology and Epigenetics, working with colleagues at the University of Washington, examined the genomes of ceratioid anglerfish species across the suborder and found that the genes encoding key components of the adaptive immune system were absent or substantially reduced across multiple lineages. The specific components missing included elements of the T-cell receptor pathway and certain antibody production pathways that in other vertebrates are central to the rejection response. The anglerfish appear to have lost the adaptive immune machinery that makes permanent tissue fusion between unrelated individuals impossible in other species. The consequence is that they can fuse without rejection. The cost of that capacity appears to be reduced immune defense against pathogens and parasites. How that trade-off has shaped the susceptibility of ceratioid anglerfishes to disease and infection is a question the researchers identified as requiring further investigation. Theodore Peach, at the University of Washington, has been the central taxonomic authority on Ceratioidae for several decades. His comprehensive monograph on the group, published in 2009, organized the more than 160 described species into their families and genera, resolved historical misidentifications that had accumulated from the practice of describing males and females as separate species, and provided the framework within which subsequent taxonomic and phylogenetic work on the group is conducted. The monograph drew on museum-type specimens held at institutions across multiple countries, many of which had not been examined against each other before. The revision it produced collapsed a number of redundant species names and established relationships between families that earlier work had left unresolved. Subsequent molecular phylogenetic analysis has modified some of those relationships, but the monograph remains the foundational reference. Across the 11 families within Ceracioidae, body forms diverge substantially. Melanocetus johnsoni, the black sea devil, has a globular dark body and an esker positioned close to the broadly gaping mouth, producing the silhouette most commonly depicted in natural history illustrations of the group. Himantolophus groenlandicus, the football fish, is heavier-bodied with an elaborate esker bearing multiple appendages and branching structures. Caulifrene jordani, the fanfin seideville, carries extremely long, filamentous extensions of the dorsal and anal fins that trail around the body and bear their own bioluminescent filaments, giving the animal a structure more reminiscent of a drifting organism than a fish in any conventional sense. Lignophrine species carry both an elysium with an esker and a highly branched, tree-like bioluminescent barbel hanging from the lower jaw, effectively operating two independent light-producing structures simultaneously. The diversity of form across the group reflects different functional solutions to the same core problem of hunting in total darkness with minimal energy expenditure. Direct observation of living ceratioid anglerfishes behaving naturally in the bathypelagic zone is rare to a degree that makes the giant squid footage discussed in the previous section seem relatively well documented. Most of what is known about the group comes from preserved trawl specimens, examination of stomach contents, and inference from anatomy. In 2020, Kirsten and Joachim Jakobsen, operating a private submersible in waters off the Azores, filmed a living Caulifrene jordani at approximately 800 meters depth. The footage showed the animal hanging nearly motionless, its long fin rays trailing around it, the bioluminescent filaments along those rays producing light in the surrounding darkness. The fish appeared undisturbed by the submersible's presence for much of the observation. It was a female. The footage represented one of the very few recordings of a ceratioid anglerfish in undisturbed natural behavior and was widely circulated through scientific and popular media. The bioluminescence of the bathypelagic zone extends far beyond the anglerfish lure. Estimates from trawl sampling data suggest that between 70% and 90% of animals living in the deep sea produce some form of bioluminescence. This makes the midnight zone, despite receiving no solar light whatsoever, a zone of substantial biological light production distributed across its resident species. That light serves different functions in different animals. Prey attraction, as in the anglerfish, predator deterrence, through sudden bright flashes that startle or blind an approaching predator. Counter-illumination, in which an animal produces light on its underside to match the faint downwelling glow from above and reduce its silhouette visibility. And communication between individuals of the same species during reproduction or aggregation. The anglerfish lure is one highly specialized application of a biological capacity shared across the majority of the zone's inhabitants. The sampling methods available for the bathypelagic zone impose significant limitations on what can be known about it. Trawl nets are hauled through the water column and retrieve whatever animals cannot escape the net or are damaged and killed by the pressure changes during retrieval. Soft-bodied animals rarely survive the journey to the surface intact, and many specimens arrive in a state that makes detailed anatomical examination difficult. Submersible observations cover a very small volume of water per dive and are limited by the depth ratings of available vehicles and the cost of operating them. The combined result is that the species inventory of the bathypelagic zone is almost certainly incomplete. The population sizes and distributions of described species are largely unknown, and the behavior of even the best-studied species is inferred from preserved specimens and the rare filmed encounter rather than from sustained direct observation. The anglerfish is the most recognizable symbol of the midnight zone and one of its least understood residents. Before we move on, if you're enjoying this analysis, please consider subscribing. It helps the channel tremendously. The next section descends further into the abyssal zone, where a creature related to the common garden pillbug has grown to the size of a football, refused food for 5 years in captivity, and waits on the seafloor for a whale to fall. Section 8. The Abyssal Zone. Giant Isopod. The abyssal zone begins at around 4,000 meters and extends to approximately 6,000 meters, where the Hadal Zone of the deep ocean trenches takes over. It covers more of the Earth's surface than all the land combined. Pressure at 5,000 meters is approximately 500 times the pressure at the surface. Temperature hovers just above freezing across the entire zone with very little variation. Light is completely absent. Food is scarce in a way that makes the mesopelagic zone above seem comparatively well provisioned. The abyssal floor is largely sediment, a deep layer of fine material accumulated from millions of years of marine snow falling from the zones above, compressed under pressure into a soft, flat substrate that stretches across the deepest parts of every ocean basin. Very little moves across it. When something does move, it tends to be patient. Bathynomus giganteus is a crustacean. It is related to the pillbugs found in garden soil, under rocks and rotting wood, the small gray animals that roll into a tight sphere when disturbed. The relationship is not superficial. Giant isopods and terrestrial pillbugs belong to the same order, Isopoda, and share the same fundamental body plan: a segmented, armored body with overlapping plates, 7 pairs of walking legs, 2 pairs of antennae, and compound eyes positioned on the sides of the head. A pillbug reaches around 2 centimeters. Bathynomus giganteus reaches 45 centimeters, occasionally beyond, and weighs up to approximately 1.5 kilograms. The scale of the difference between a pillbug on a garden path and a giant isopod on the abyssal floor is one of the more striking examples of deep-sea gigantism documented in any invertebrate group. Deep-sea gigantism is the tendency of animals living in the deep ocean to grow considerably larger than their shallow water or terrestrial relatives. It is documented across multiple phyla and has been observed in isopods, amphipods, sea spiders, and various other invertebrate groups represented in the deep sea. The explanation for it is not fully settled. Several mechanisms have been proposed individually and in combination. Cold temperatures reduce metabolic rates, potentially allowing organisms to continue growing over longer lifespans than would be possible in warmer water. Reduced predation pressure in the deep sea may remove the selective constraints on body size that operate in shallower zones with more predators. The nature of food availability in the deep sea, long periods of scarcity, interrupted by large episodic inputs from whale falls and other major sinking events, may favor larger body size because a larger animal can store more energy between feeding events. Oxygen availability has also been proposed as a contributing factor. None of these explanations has been demonstrated as the primary driver across all taxa, showing the pattern, and the relative contribution of each likely varies between groups and between ocean regions. Bathynomus giganteus was first formally described in 1879 by the French zoologist Alphonse Milne Edwards from specimens collected in the Gulf of Mexico. The discovery placed a deep-sea animal of unexpected size into the scientific literature at a moment when the systematic exploration of the deep ocean was just beginning to generate specimens for examination. The HMS Challenger expedition, which ran from 1872 to 1876 and is generally treated as the founding expedition of modern oceanography, had established that complex animal life existed at abyssal depths, which had previously been considered too hostile for any biology. The discovery of Bathynomus giganteus, a few years after Challenger's findings, added to the accumulating picture of the deep ocean as populated by a fauna that was not simply a sparse extension of shallower life, but a distinct assemblage of animals with their own size ranges, body plans, and adaptations. The specimen Milne Edwards described was a female, and the species was immediately recognized as the largest isopod then known to science, a distinction it still holds today. The body of Bathynomus giganteus is covered in a rigid, segmented exoskeleton of overlapping plates that provide protection against the ambient pressure and against physical damage. The animal can roll itself into a tight sphere by tucking its legs beneath the body and bringing the tail section around to meet the head, sealing itself into a near-complete ball that exposes only the hard dorsal plates. This defensive posture is the same one used by pillbugs when threatened, and it is effective against a range of potential predators. The compound eyes are large relative to the body, positioned laterally to give a wide field of view, and are reflective in a way that produces a distinctive eye shine in artificial light. Despite this visual apparatus, the deep-sea environment in which the animal lives is completely lightless, and the compound eyes of Bathynomus giganteus are almost certainly more sensitive to detecting bioluminescent flashes from other organisms than to processing detailed visual information. The antennae and chemoreceptors distributed across the body surface are the primary sensory tools for locating food in the dark. The feeding behavior of Bathynomus giganteus is shaped entirely by the economics of scarcity and abundance in the abyssal zone. The animal is a scavenger. It does not actively hunt living prey in any meaningful sense. It feeds on material that arrives at the seafloor from above, primarily the bodies of dead fish, squid, and marine mammals. When food becomes available, Bathynomus giganteus feeds rapidly and extensively. Observations from baited camera deployments and from baited trap collections have documented the animals arriving at food sources in large numbers and consuming significant quantities of material in short periods. The mouth is equipped with 4 sets of mouthparts working together to tear and process food, and the feeding rate, when food is present, is fast relative to the animal's otherwise very slow pace of movement. After feeding, the animal enters what researchers describe as a state of very low activity that may persist for extended periods. Its metabolic rate already low from the cold, and pressure adjusted downward further by the demands of digesting a large meal and waiting for the next opportunity. The most extensively documented example of the giant isopod's capacity for fasting comes from a specimen held at Toba Aquarium in Mie Prefecture, Japan. The animal arrived at the aquarium in 2007 and initially accepted food. On January 2, 2009, it ate for the last time. It then refused every food item offered to it for the remainder of its time at the facility. Staff attempted to feed it mackerel, squid, and other items across the following years. The animal declined all of them. It remained alive, active enough to move when handled, visibly responding to its environment. It died on February 14, 2014. The interval between its last recorded meal and its death was 5 years and 43 days. The aquarium documented the fast in detail and published information about it, and the case was reported in science media and natural history publications as a documented record of food refusal duration in a captive animal. Whether the Toba specimen's fast reflected a physiological condition specific to that individual, a response to captivity, or a demonstration of the species' genuine capacity for prolonged fasting under abyssal-like conditions of low activity, is not established. What was established was the duration, because it was logged continuously from the date of the last meal to the date of death. The physiological basis for this capacity involves a metabolic rate that is extraordinarily low even by the standards of cold water invertebrates. Abyssal conditions, the cold, the pressure, and the scarcity of food exert strong selective pressure toward energy conservation. Bathynomus giganteus moves slowly. Its heart rate is low. Its oxygen consumption per unit body mass is a fraction of what would be expected from a similarly sized animal in warmer, shallower water. The energy stored from a large feeding event can be distributed over an extended period of very low expenditure, sustaining the animal through intervals between food arrivals that would be fatal to an animal with a higher metabolic rate. The trade-off is that growth is slow and reproduction is infrequent. But in an environment where food cannot be reliably predicted, those trade-offs appear to be worth making. Whale falls are among the most significant food sources in the abyssal and deep-sea zones. When a large whale dies in the open ocean, its body sinks. The descent through the water column takes hours. When it reaches the seafloor, it initiates a succession of ecological stages that researchers studying deep-sea whale fall ecology have documented in detail at several sites, primarily through repeated submersible dives to known locations. The initial stage, called the mobile scavenger stage, involves large scavengers arriving quickly, within hours to days of the carcass reaching the floor. Sleeper sharks, hagfish, and large crustaceans, including giant isopods, have been documented at whale falls during this stage, feeding rapidly on the soft tissue. A single large whale carcass can provide enough organic material to sustain a complex community of organisms for decades through successive ecological stages as different groups of species process different components of the remains. The bone-eating worms of the genus Osedax, discovered in 2002, consume the lipid-rich whale bones during later stages of the succession, releasing nutrients that support bacterial communities at the site. The whale fall is, in the context of the abyssal floor, a sudden and enormous input of energy into an otherwise nutrient-poor environment, and the fauna that has evolved to exploit it is correspondingly specialized. The reproductive biology of Bathynomus giganteus involves internal fertilization and direct development, with no free-swimming larval stage of the kind seen in many marine invertebrates. Females brood their eggs in a specialized pouch called the marsupium on the underside of the body, and the young emerge as fully formed miniature versions of the adult, called mancae, already capable of independent movement and feeding. The brood sizes observed in collected specimens have been relatively small compared to the reproductive output of many marine invertebrates. This low reproductive rate, combined with slow growth and extended lifespans, means that Bathynomus giganteus populations respond slowly to changes in the environment and would be slow to recover from significant mortality. Direct observation of reproduction in the wild has not been achieved, and most of what is known about the reproductive biology comes from examination of collected females carrying eggs or mancae, supplemented by observations of the few individuals that have survived and bred in captivity. The genus Bathynomus contains multiple species beyond Giganteus, distributed across different ocean regions and depth ranges. Bathynomus doedalini, found in shallower waters of the Indo-Pacific, is considerably smaller than Giganteus and occupies a different ecological niche. Bathynomus jamesi, described from deep water in the Indian Ocean, is among the more recently named species in the genus. Molecular analysis has clarified the relationships between species in some cases, while revealing that what had been treated as a single species was in some instances multiple distinct lineages. The taxonomy of the genus has been revised several times as new collections have become available, and it is likely that additional species remain to be formally described from deep-sea collections that have not yet been fully processed. Several individuals of Bathynomus giganteus and related large Bathynomus species have been kept in public aquariums and research facilities, and their behavior in captivity has provided some of the most detailed observational records available for the group. Beyond the toba specimen, captive animals have been documented exhibiting the rolling defensive posture, responding to light stimuli with avoidance behavior consistent with the sensitivity of their compound eyes, and occasionally accepting food in quantities and at intervals that loosely parallel what might be expected from the episodic feeding pattern in the wild. The difficulty of maintaining deep-sea animals in captivity relates partly to pressure and temperature requirements, and partly to the challenge of providing appropriate food at appropriate intervals. Animals brought rapidly from depth experience pressure changes that damage tissue and circulatory systems. Successful captive maintenance of Bathynomus has generally involved collection from shallower parts of the species' depth range and gradual acclimatization, rather than collection from the deepest parts of the abyssal habitat. The abyssal floor across which Bathynomus giganteus and its relatives move is not uniform. Sediment plains, abyssal hills, mid-ocean ridges, and the scattered hard substrates provided by manganese nodules, potato-sized concretions of metal oxides that form slowly on the sediment surface, create a varied physical landscape within the zone. Manganese nodules have attracted commercial interest for several decades because they contain concentrated deposits of metals, including manganese, nickel, copper, and cobalt. Proposals for abyssal mining of these nodule fields have raised concerns among deep-sea ecologists because the fauna living on and around the nodules, including isopods, polychaete worms, and various other invertebrates, has taken millions of years to colonize the slow-growing nodule surfaces. Disturbance of the sediment by mining equipment would destroy that community and, given the slow pace of biological processes at abyssal depths, recovery would not occur on any human timescale. Studies of previously disturbed abyssal sediment, including sites disturbed by research activities in the 1970s and revisited decades later, have found that recovery of the biological community was minimal even after 26 years, supporting the concern that abyssal mining would produce effectively permanent local extinction of the community at each mined site. The giant isopod is not the only animal in the abyssal zone that demonstrates the adaptations of that environment in a striking way. Sea cucumbers, holothurians, dominate the abyssal plain by biomass in many regions, moving slowly across the sediment surface and processing the organic material within it. Xenophyophores, single-celled organisms that build cases from sediment particles and reach sizes of up to 20 centimeters, making them among the largest individual cells known to science, are common on abyssal sediment surfaces. Polychaete worms thread through the sediment in numbers that make the abyssal floor, despite its apparent bleakness from above, a densely populated habitat at the scale of the organisms that live in and on the sediment layer. Bathynomus giganteus moves through this community as one of its largest and most mobile members, crossing sediment plains between food sources, arriving at whale falls in numbers that suggest some chemosensory ability to detect food from a distance in the dark, and waiting between arrivals with a patience that is simply metabolic necessity expressed in behavior. Before we move on, if you're enjoying this analysis, please consider subscribing. It helps the channel tremendously. The next section descends to the seafloor itself, not the open abyssal plain, but the volcanic cracks, where superheated water vents into the cold dark, and where the most heat-tolerant complex animal, documented by science, builds its tubes directly on the chimney walls. Section 9. The Hydrothermal Vent Zone. Pompeii worm. In February 1977, the research submersible Alvin descended to 2,500 meters near the Galapagos Rift, a spreading zone on the Pacific seafloor where two tectonic plates pull apart from each other. The scientists aboard were geologists and geochemists, primarily interested in the warm water they expected to find venting from cracks in the seafloor as a consequence of the heat generated by the spreading plates below. They found the warm water. They also found, clustered around the vents in densities that made the surrounding seafloor look barren by comparison, a community of living organisms unlike anything previously documented at that depth. Giant clams the size of dinner plates, crabs moving through water that should, by the prevailing understanding of deep-sea biology, have contained almost nothing worth eating. Tube worms, white-cased and tipped with red plumes, rising from the seafloor in thickets, dense enough that the submersible's lights could not reach through them to the rock below. The scientists transmitted a message to the surface ship above. The warm water is there, and there is life there too. The team on the surface, receiving reports from the seafloor in real time, understood immediately that the finding was significant beyond anything the dive had been planned to produce. The 1977 Galapagos Rift discovery did not just add a new habitat to the known marine catalogue, It overturned a foundational assumption about life on Earth. Before that dive, the organizing principle of all known ecosystems on Earth was photosynthesis. Plants, algae, and photosynthetic bacteria capture energy from sunlight and convert it into organic material. Everything else in the food web, from herbivores to apex predators, runs on that converted solar energy, either directly or through the chain of organisms that have eaten organisms that ate the photosynthesizers. The deep sea was known to support life, but that life was understood to depend ultimately on organic materials sinking from the sunlit surface, meaning the deep sea was photosynthetically subsidized, even if indirectly. The vent community at the Galapagos Rift was not. The bacteria at the base of its food web were chemosynthetic, deriving energy not from light, but from the chemical reaction of oxidizing hydrogen sulfide venting from the seafloor. No sunlight reached them. No sunlight was required. The vent ecosystem ran entirely on chemical energy from the Earth's interior, and it was productive enough to support large, complex, long-lived animals in densities that rivaled anything in the sunlit ocean above. The geological mechanism producing hydrothermal vents is consistent across all known vent fields, which have since been discovered along mid-ocean ridges in every ocean basin. Cold seawater percolates through cracks and fissures in the seafloor, sinking toward the heat source below. As it descends, it heats. At depth near the magma that drives the spreading, water temperatures can reach 400 degrees Celsius, far above the boiling point at surface pressure, but kept liquid by the enormous pressure of the overlying water column. The superheated water dissolves minerals from the surrounding rock, loading itself with sulfides, metals, and other compounds. It then rises through the rock and vents back into the seafloor through openings in the seafloor surface. When the superheated, mineral-rich water contacts the near-freezing ambient deep-sea water, the dissolved minerals precipitate rapidly, building chimney-like structures, called black smokers when the mineral mix is iron and sulfide compounds, and white smokers when the temperature is lower and the mineral composition different. A black smoker chimney can reach several meters in height and vent water at temperatures measured in excess of 350 degrees Celsius, producing a dark plume of precipitating minerals that looks, from a submersible viewport, like thick black smoke rising from the seafloor. The organisms living on and around these chimney structures exist in one of the most physically extreme environments documented anywhere on Earth. The temperature gradient around a single vent chimney runs from above 300 degrees at the vent opening to around 2 degrees Celsius in the ambient deep-sea water a few meters away. The water is acidic. It is laced with hydrogen sulfide, which is toxic to aerobic organisms above trace concentrations. The pressure at typical vent depths of 2,000 to 3,000 meters is between 200 and 300 times the surface pressure. The vent community thrives in all of this, concentrated along the thermal gradient at the specific temperature and chemistry ranges that its members are each adapted to tolerate. Among the vent fauna documented in the years following the 1977 discovery, one animal was identified in 1980 during dives by French and American research teams on the East Pacific Rise that established itself as the most heat-tolerant complex animal yet known to science. The Pompeii worm, described formally by French marine biologists Daniel de Bruyere and Lucien Laubier, was named after the Roman city buried by Vesuvius because the grey, ash-like bacteria covering its back reminded the researchers of the volcanic deposits that had preserved Pompeii. The comparison was apt in more ways than the visual one. Like Pompeii, the worm lives in the immediate proximity of volcanic heat that would destroy most other complex life, and persists there through adaptations that are not yet fully understood. Alvinella pompeiana, the formal name honoring both the submersible Alvin and the Pompeii Association, is a polychaete worm, a member of the same broad group that includes the common earthworm's marine relatives. It reaches around 13 centimeters in length. It builds a papery tube attached to the surface of active hydrothermal vent chimneys. The tube extends from the chimney wall outward into the surrounding water. The worm lives inside the tube with its head end at the opening, exposed to the cooler water outside, and its tail end deeper in the tube, closer to the chimney wall. The temperature at the head end, where the worm's most metabolically sensitive tissues are located, is around 22 degrees Celsius. The temperature at the tail end, based on measurements taken by researchers inserting temperature probes into occupied tubes during submersible dives, reaches into ranges that have been reported in the scientific literature at up to 80 degrees Celsius. That temperature figure, 80 degrees Celsius at the tail end of an inhabited worm tube, is the most extreme thermal tolerance documented for any complex multicellular animal. A temperature of 80 degrees Celsius is close to the point at which most proteins begin to denature and lose function irreversibly. The cells of most complex animals, including humans, suffer rapid and catastrophic damage at temperatures above around 45 degrees Celsius. The Pompeii worm's cells at the tail end of its tube are functional at temperatures approaching twice that threshold. The thermal gradient experienced across a single worm, from approximately 22 degrees at the head to potentially 80 degrees at the tail, occurs across a tube length of roughly 10 centimeters, making it one of the steepest thermal gradients endured by any known animal. The 80-degree figure has been the subject of scientific debate since the measurements were first published. Some researchers questioned whether the temperature probes inserted into worm tubes during submersible dives were measuring the temperature at the precise location of the worm's tissue, or the temperature of the hottest water in the tube, which may not be where the worm actually positions itself within the tube during normal occupation. The concern was that the worm might retreat toward its tube opening when the temperature becomes extreme, meaning the hottest recorded tube temperature may not represent the temperature the worm routinely tolerates at its tail end. More conservative interpretations of the data have suggested that the worm's functional thermal tolerance may be closer to 55 to 60 degrees Celsius, which remains the highest documented for any complex animal but is more consistent with what is known about protein stability at high temperatures. The debate reflects genuine methodological difficulty. Measuring the microenvironment experienced by a specific body part of a small worm inside a tube on an active hydrothermal chimney at 2,000 meters depth, without disturbing the worm or the tube, is not a straightforward instrumentation problem. The upper temperature figure remains in the literature as the most commonly cited. Its precise accuracy is contested. The extraordinary nature of the animal's thermal tolerance is not. The fleece of bacteria covering the worm's dorsal surface is one of the most closely studied aspects of its biology, because it sits at the center of several hypotheses about how the worm survives its thermal environment. The bacterial layer is dense and visually distinctive, a pale gray-white covering across the worm's back that gave the Pompeii comparison its initial force. Molecular analysis of the bacterial community has identified the dominant members as filamentous bacteria related to Epsilon proteobacteria, a group widely represented in hydrothermal vent and other chemosynthetic environments. The relationship between the worm and its bacterial layer is mutualistic rather than parasitic. The worm secretes mucus from glands on its back that the bacteria feed on, providing them with organic material. What the bacteria provide to the worm has been the subject of multiple proposed mechanisms. One hypothesis is nutritional. The bacteria may supplement the worm's diet by fixing carbon through chemosynthesis and providing organic compounds that the worm absorbs through its skin. Another hypothesis is thermal insulation. The dense bacterial layer may act as a biological insulating mat, reducing the rate of heat transfer to the worm's dorsal tissue. A third possibility, not mutually exclusive with the others, is that the bacteria help detoxify hydrogen sulfide in the worm's immediate microenvironment, reducing the chemical stress on the worm's tissues. Research has provided support for elements of multiple hypotheses without fully resolving which function is primary. The bacterial community on the worm's back is functionally significant. The precise mechanism of its significance to the worm's survival remains an active area of investigation. The biochemical adaptations of Alvinella pompeiana at the cellular level have been examined through molecular biological techniques applied to tissue samples collected during submersible dives. Heat shock proteins, molecular chaperones that help other proteins maintain correct folding under thermal stress, are present in elevated concentrations in Pompeii worm tissue compared to related polychaete worms living at ambient deep-sea temperatures. Heat shock proteins are produced by virtually all organisms in response to elevated temperature, but the baseline levels in Pompeii worm tissue suggest a chronic rather than acute response, meaning the worm lives in a state of continuous heat stress management rather than responding to occasional thermal challenges. The lipid composition of the worm's cell membranes has also been analyzed, and found to differ from closely related worms in ways consistent with maintaining membrane fluidity and function at higher temperatures. Cell membranes that work well at typical biological temperatures become increasingly rigid as temperature rises, and the Pompeii worm's membrane lipids appear to be compositionally adjusted to remain functional across a wider temperature range than those of its non-vent relatives. Daniel Desbruyères, working at the French oceanographic research organization IFREMER, continued studying vent fauna and Alvinella pompeiana, specifically across a career spanning several decades after the initial description. Desbruyères made repeated dives to the East Pacific Rise vent fields aboard both Alvin and the French submersible Nautil, contributing to the understanding of Pompeii worm biology, tube structure, bacterial community composition, and the broader ecology of the vent community in which the worm lives. His work, and that of colleagues across French, American, and international institutions, established the East Pacific Rise vent fields as the most intensively studied hydrothermal vent systems in the world, providing the baseline against which vents discovered subsequently in other ocean basins have been characterized. The broader vent community that surrounds Pompeii worm colonies on the East Pacific Rise chimneys includes organisms that are in some cases equally extraordinary in their adaptations, even if none has been documented tolerating temperatures as high as Alvinella pompeiana. The giant tube worm Riftia pachyptila, formerly described by Meredith Jones in 1981, from specimens collected during post-Galapagos dives, reaches up to 2 meters in length and lives in a white tube attached to the seafloor near vents. As an adult, Riftia has no mouth and no digestive system. It feeds entirely through an internal organ called the trophosome, which is packed with chemosynthetic bacteria. The bacteria oxidize hydrogen sulfide delivered to them through the worm's blood and produce organic compounds that sustain the worm. The worm's bright red plume, the gill-like structure that extends from the tube opening, is rich in a specialized hemoglobin that carries both oxygen and hydrogen sulfide simultaneously to the trophosome bacteria without the sulfide poisoning the hemoglobin in the process, a chemical feat that required dedicated study to understand. The Yeti crab, Kiwa hirsuta, discovered in 2005 at a vent field on the Pacific Antarctic Ridge by a team led by Michel Segonzac of IFREMER and Robert Vriegenhoek of the Monterey Bay Aquarium Research Institute, farms bacteria on its own body rather than harboring them internally. The crab's claws are covered in dense, hair-like setae that host mats of chemosynthetic bacteria. The crab waves its claws in rhythmic motions near the vent flow, apparently maximizing the bacteria's exposure to the chemical-rich vent water, and then scrapes the bacterial mats into its mouth with specialized mouthparts. The discovery of a crab that has evolved agricultural behavior applied to its own extremities added another layer to the already unusual picture of vent community ecology. The vent shrimp Rimicaris exoculata, abundant at hydrothermal vents along the mid-Atlantic ridge, is effectively eyeless in the conventional sense, lacking the compound eyes found in most shrimp. Instead, it has 2 large photoreceptive organs on its back, beneath a transparent section of the carapace. These organs are connected to the optic lobes of the brain and are sensitive to very low-intensity light. The leading hypothesis is that they detect the faint glow of thermal radiation emitted by the superheated vent water, allowing the shrimp to navigate toward and away from the vent flow without approaching close enough to be cooked. The vents themselves emit detectable radiation in wavelengths visible to sensitive photoreceptors, even though no sunlight reaches the depth. Rimicaris exoculata appears to see by the light of the Earth's own heat. The vent fields of the East Pacific Rise, where Alvinella pompeiana was discovered, are among the most geologically active hydrothermal systems known. Individual vent chimneys can be ephemeral on geological timescales, becoming inactive as the flow beneath them shifts and new vents open nearby. The vent community must colonize new sites as old ones die, and the mechanisms by which larvae of vent species disperse through the deep ocean to find new active vents are an active area of research. Pompeii worm larvae, like those of most vent species studied, are planktonic for a period after hatching, drifting through the water column before settling. How they locate an active vent from the surrounding deep-sea water, and over what distances dispersal is effective, shapes the connectivity between vent populations across the ridge system. The discovery of chemosynthetic ecosystems at hydrothermal vents extended in subsequent decades to cold seeps, where methane and hydrogen sulfide leak from the seafloor without the thermal component of vent systems, supporting communities structurally similar to vent fauna but driven by different chemical energy sources. It extended to whale falls, discussed in the previous section, where the organic content of a decomposing whale body supports chemosynthetic bacteria and a succession of dependent organisms. The recognition that photosynthesis is not the only foundation for complex animal life has had implications that reach beyond marine biology into the search for life in environments where sunlight is absent, including the subsurface oceans beneath the ice shells of Jupiter's moon Europa and Saturn's moon Enceladus, where hydrothermal activity has been proposed as a potential energy source for biology, if biology exists there. The Pompeii worm living in its tube on a chimney wall on the East Pacific Rise is a demonstration, documented and measured, that complex multicellular life can persist in conditions that resemble in some ways what those subsurface oceans may contain. Before we move on, if you're enjoying this analysis, please consider subscribing. It helps the channel tremendously. The final section descends to the deepest place on Earth, into the Hadal Trenches below 6,000 meters, where crustaceans swarm the seafloor in numbers that surprised every research team that sent traps down. Section 10. The Hadal Zone. Giant amphipods. On January 23, 1960, the bathyscaphe Trieste settled onto the floor of the Challenger Deep at a depth of approximately 10,900 meters. The two men inside, Swiss oceanographer Jacques Piccard and United States Navy Lieutenant Don Walsh, had spent nearly 5 hours descending from the surface. When they arrived, they looked through the viewport into the sediment, disturbed by their landing, and reported seeing a small flatfish resting on the bottom. The claim was debated for years afterward, with some researchers suggesting the animal was more likely a sea cucumber than a fish. Walsh maintained the identification throughout his life. Whether the animal was a fish or not, the observation established that something was alive at the deepest point on Earth, and that the Hadal Zone, named for the Greek god of the underworld, was not the sterile void that some researchers had assumed it must be. The Hadal Zone is defined as the ocean below 6,000 meters. It exists almost entirely within the ocean trenches, the long, narrow depressions in the seafloor formed where one tectonic plate slides beneath another in a process called subduction. There are roughly 37 Hadal trenches identified across the world's ocean basins. The deepest is the Challenger Deep in the Mariana Trench, located in the western Pacific Ocean, which reaches a confirmed depth of approximately 10,928 meters, according to measurements conducted during the Five Deeps expedition in 2019. Pressure at that depth exceeds 1,000 times the pressure at the surface. Temperature is just above freezing. Light is completely absent. The seafloor is soft sediment, accumulated from millions of years of particles settling from above, compressed into a pale, flat substrate that appears, in submersible footage, almost featureless. The Five Deeps expedition, funded by American explorer Victor Vescovo and conducted aboard the research vessel DSSV Pressure Drop using the submersible DSV limiting factor, reached the bottom of the deepest point in each of the 5 ocean basins between 2018 and 2019. When Vescovo reached the floor of the Challenger Deep in April 2019, setting a new depth record for a crewed descent, the team found the expected sediment, the expected darkness, and the expected cold. They also found a plastic bag and sweet wrappers on the seafloor. The items were documented, photographed, and reported. The finding was not the first detection of human-produced debris at Hadal depths, but it was among the most widely publicized, because the juxtaposition of a candy wrapper and the deepest place on Earth communicated something about the reach of human pollution that scientific papers had been documenting in less visually immediate terms for years before that dive. The Hadal trenches, because of their geometry and their position at the bottom of the ocean, function as sediment traps. Particles falling through the water column from above, marine snow, dead organisms, organic debris, concentrate in the trench as surrounding topography funnels, settling material toward the lowest point. This accumulation gives Hadal sediments organic content higher than would be expected from the open abyssal plain at comparable depths. And that organic richness supports a fauna denser than many researchers anticipated, before systematic sampling of Haddle zones began. The animals most consistently found in Haddle traps and cameras, arriving in numbers large enough to cover a bait parcel within hours of it reaching the floor, are amphipods. Amphipods are crustaceans, laterally compressed, shrimp-like in general form, with two pairs of antennae and bodies divided into clearly articulated segments. In most marine environments, they are small animals, rarely exceeding a few centimeters. In the Haddle zone, certain species grow to dimensions that place them among the most striking examples of deep-sea gigantism in any invertebrate group. Alicella gigantea, described formally from specimens collected in deep-sea trawls in the late 19th and early 20th centuries and subsequently recovered from Haddle trap deployments, reaches lengths exceeding 34 centimeters in the largest documented specimens. Eurythanes gryllus, another large-bodied species found across a range of deep-sea and Haddle environments, grows to comparable dimensions. Hirondelia gigas is the species most consistently and abundantly recovered from Haddle trenches specifically, including the Mariana Trench, and while it does not reach the extreme dimensions of Alicella gigantea, it is present at Haddle sites in numbers that make it the dominant animal at many trapping stations. The arrival of Haddle amphipods at baited landers, deployed to trench floors, has been documented in camera footage from multiple research programs and is one of the more viscerally striking images to emerge from Haddle research. Hundreds of individuals, sometimes reaching densities of thousands within camera range of a single bait parcel, converge within hours of the lander reaching the bottom. They move with a speed and coordination that contradicts the popular image of the deep sea as a place of extreme lethargy. The animals arrive from the surrounding sediment, drawn by the chemical signal of the bait dispersing through the water. They cover the bait parcel, feed rapidly, and disperse. The feeding event, observed in full from descent of the lander to recovery, unfolds on a timescale of hours rather than the days or weeks one might expect from animals living in an environment of chronic food scarcity. The capacity for rapid, large-scale feeding is consistent with the episodic nature of food availability in the Haddle zone. For most of the time, the trench floor offers marine snow and the organic material within the sediment, a diffuse and low-energy food source. Occasionally, a whale falls, a fish dies and sinks, a bait parcel arrives on a research lander. When it does, the ability to locate it quickly and consume large quantities rapidly is a direct adaptive advantage. Hirondelia gigas and related Haddle amphipods have evolved not only behavioral responsiveness to food events, but digestive capacity sufficient to process large meals quickly. Analysis of digestive enzymes in Hirondelia gigas found that the species produces cellulase, an enzyme capable of breaking down cellulose, the structural material in plant cell walls. Wood has been recovered from the floor of the Mariana Trench, carried into the deep ocean by currents from rivers and coastal areas, and eventually sinking to Haddle depths. The ability of Hirondelia gigas to digest cellulose means it can extract energy from this wood, a food source unavailable to most marine animals, giving it access to an organic resource present in Haddle sediment that other Haddle species cannot use. The formal study of Haddle fauna has been transformed over the past 2 decades by the development of full ocean depth landers, free-fall instruments that carry baited traps and cameras to trench floors without a tether connecting them to the surface. Earlier, Haddle sampling relied primarily on weighted trawl nets and dredges hauled across the seafloor, which recovered specimens but provided limited behavioral data and could not consistently sample the deepest trench zones. Landers descend freely, triggered to release their ballast weights by an acoustic signal from the surface ship, and float back to the surface for recovery, carrying the animals trapped and the footage recorded during their time on the bottom. Alan Jameson, a marine scientist who worked at Newcastle University and subsequently at the University of Western Australia, has been among the most prolific users of this technology for Haddle research, conducting lander deployments at trench sites across the Pacific, Atlantic, and Indian Oceans, and accumulating the largest systematic dataset on Haddle amphipod distribution and biology produced by any single research program. It was Jameson's program that produced the finding that, beyond the biological extremity of the Haddle fauna, received the most widespread attention. Research published in Nature Ecology and Evolution in February 2017 led by Jameson and co-authored by researchers including Johanna Weston, analyzed the tissue of amphipods collected from the Mariana Trench and the Kermadec Trench, a Haddle Trench northeast of New Zealand, reaching approximately 10,000 meters. The analysis found persistent organic pollutants in the tissue of amphipods from both trenches at concentrations that were not trace-level contamination, but substantial accumulation. Polychlorinated biphenyls, known as PCBs, and polybrominated diphenyl ethers, flame-retardant compounds known as PBDEs, were detected in the amphipod tissue from both sites. The PCB concentrations in Mariana Trench amphipods exceeded those measured in amphipods from Suruga Bay in Japan, described by the researchers as one of the most polluted industrial marine environments in the northwestern Pacific. The Kermadec Trench samples showed similarly elevated levels. The most remote, most isolated, deepest fauna on Earth was carrying industrial chemical contamination in its tissue at concentrations higher than animals living in heavily polluted coastal waters. PCBs are synthetic industrial chemicals used extensively in electrical equipment, plastics, and other industrial applications from the 1930s onward. They are extremely persistent, meaning they resist biological and chemical breakdown over long periods. They are lipophilic, meaning they bind preferentially to fatty tissue rather than water. The United States banned the manufacture of PCBs in 1979. The Stockholm Convention on Persistent Organic Pollutants, a global treaty targeting PCBs and related compounds, was adopted in 2001 and entered into force in 2004. Despite these restrictions, the total volume of PCBs produced over the decades of their use is enormous, and the compound's persistence means that material released into the environment before and after regulatory restrictions continues to circulate. PBDEs, used as flame retardants in electronics, furniture foam and textiles, followed a similar trajectory, with their hazardous persistence recognized and restrictions introduced progressively in different jurisdictions from the early 2000s onward. The mechanism by which these compounds reach the floor of the Mariana Trench is a consequence of the same particle settling dynamics that give Hadal sediments their elevated organic content. Persistent organic pollutants in the ocean bind to organic particles. Those particles, along with everything else, become marine snow. The snow falls. At Haddle depths, the trench geometry concentrates the settling material. The compounds accumulate in the sediment and in the tissue of the organisms that eat that sediment and the organic material within it. Because the compounds are lipophilic, they concentrate in fatty tissue as they move up the food chain, a process called biomagnification. An amphipod eating contaminated organic particles accumulates a higher concentration of the pollutant than the particles themselves contained. An animal that eats many contaminated amphipods accumulates higher concentrations still. The Haddle Trench, functioning as a sediment trap, also functions as a pollutant trap, accumulating compounds from an enormous area of ocean surface above it and concentrating them in the sediment and ultimately in the fauna. The Jameson study described the trenches as potential sinks for persistent organic pollutants on a global scale, receiving contamination carried from industrialized regions thousands of kilometers away by ocean currents and atmospheric deposition. A subsequent study, published in 2019 and led by researchers including Jameson, examined amphipods from 6 of the world's deepest ocean trenches for the presence of microplastics, particles of plastic below 5 millimeters in size, produced by the breakdown of larger plastic items and by the direct release of plastic pellets and fibers from industrial and consumer products. Microplastics were found in amphipods from all 6 trenches sampled, including the Mariana Trench, the Kermadec Trench, and the New Hebrides Trench. The particles were ingested, present in the digestive tracts of the animals. As with the persistent organic pollutant finding, the detection of microplastics at Hadal depths in Hadal fauna demonstrated that the distance and depth of the Hadal zone from human activity provides no isolation from the physical and chemical products of that activity. Plastic produced at the surface finds its way to the deepest floor and into the bodies of the animals living there. The Hadal snailfish, in the family Liparidae, is the deepest living fish confirmed by science. Snailfish are found across a range of deep-sea environments and have repeatedly been recorded, setting depth records at Hadal trapping stations. In 2023, a team from the University of Western Australia and Tokyo University of Marine Science and Technology, using baited camera landers, filmed a snailfish at 8,336 meters in the Japan Trench, the deepest observation of a fish ever recorded at that time. The animal was small, pale, and gelatinous in appearance, moving through the frame of the camera for several seconds before disappearing. The depth at which it was filmed places it well within the Hadal zone and requires physiological adaptations to pressure that are distinct from those of fish living even a few hundred meters shallower. Hadal fish maintain cellular function under extreme pressure through elevated concentrations of trimethylamine oxide, a compound that stabilizes proteins against the deforming effects of pressure on molecular structure, a mechanism shared with other deep-sea fish but expressed at hadal depths in concentrations above those found in mesopelagic or abyssal species. The deepest hydrothermal vents known are found in Hadal or near Hadal environments. The Bibi vent field on the Mid-Cayman Rise in the Caribbean, discovered in 2010 by a research team using the remotely operated vehicle ISIS, is located at approximately 4,960 meters, making it the deepest known hydrothermal vent system at the time of its discovery. The vent fluid temperatures measured there exceeded 490 degrees Celsius, the highest vent temperatures then recorded. Vent communities at that site share some taxonomic affiliations with vent faunas from the Pacific, but also include species unique to the site, consistent with the isolation of the Caribbean basin from Pacific vent fields. The discovery extended the known range of chemosynthetic vent ecosystems into a basin previously unsampled at those depths, and added to the picture of hydrothermal activity as a globally distributed seafloor phenomenon rather than one restricted to specific ridge systems. The 9 zones traversed across this script, from the tidal edge where the box jellyfish hunts in the warm surface shallows, to the sediment floor of the Challenger Deep, where amphipods swarm a bait parcel in the dark, represent a vertical span of nearly 11 kilometers. The ocean contains all of them simultaneously, stacked on top of each other, each with its own pressure, its own temperature, its own light regime, its own community of organisms shaped by those conditions over millions of years. The animals in each zone are not extreme in the way the word is typically used, implying something outside the normal range of experience. They are normal, within the context of the environment they inhabit. The box jellyfish navigating by light filtered through mangrove canopy, the anglerfish carrying fused males on her body through permanent darkness, the Pompeii worm with its tail in water approaching boiling point while its head remains in the cold, the amphipod in the Mariana Trench carrying industrial chemicals in its tissue from a world it has no access to and no knowledge of. All of them are doing what their biology has shaped them to do, in the place their lineage has occupied long before there were words for any of it. If you enjoyed this breakdown, make sure to subscribe. Thanks for watching, and I'll see you in the next one.